Hello. Thank you. All right. So, hi, good afternoon, everyone. My name is Adam Megebrecht. I'm here with my friend Felipe Ornjuela Espina. I apologize. It's the best I can do. So this afternoon, I hope you're all enjoying your digestion and the poster session. So we're going to do some data analysis, which is going to be a lot of fun. So we're going to start off with an invited talk by Paula Pinti. It's going to talk about mobile brain imaging with fNIRS, opportunities and methodological challenges in toddlers.
So hi everyone, thanks Adam for the introduction and for the opportunity of giving this presentation today. So in my talk, I will be discussing a little bit the process that we are going through in setting up a new lab where we want to monitor brain activity from freely moving toddlers with mobile fNIRS. So just to give you a little bit of background, just a few months ago, we have opened our new lab, which is called the Toddler Lab, as you can see in the picture there on the left. And the goal of this lab is to basically, we want to study the brain and cognitive development in toddlers and preschoolers from 3 to 6 years old in naturalistic setting. So we want children to be able to move freely in a more naturalistic situation. So in the main building, so the Toddler Lab, we have many different laboratories that resemble real-world settings. So we have, for example, a home lab that looks like a living room. We have a preschool lab that looks like a classroom, but what it is more exciting for me is our virtual reality cave lab. So the cave is basically a little room, like a cube, where we can project any virtual scenario that we want on the front, on the side walls, and on the front wall. And children are able to move around and walk around the space, but also interact with the virtual space and the virtual objects by different means, for instance, with motion tracking or eye tracking. So thanks also to the new hardware developments, we have available wireless and wearable fNIRS systems, which means that we are able to monitor brain activity also while people are freely moving in the space. So in this presentation today, I will be talking about how we are trying to achieve what is called neuroscience of the everyday world on toddlers. So we'll be discussing these 3 main topics and present some unpublished preliminary data that we are currently collecting at the moment. So first of all, when we first opened the Toddler Lab, we started by getting the fNIRS system to be as child-friendly as possible, so comfortable and lightweight so that children can move around the space. For example, we often use a little backpack where we host the main units of the NIRS device so that they can move freely, but ultimately we want to be able to get robust data out of it. So we start by running our first pilot experiment with wearable NIRS on toddlers, and also to get kind of an idea of what type of data we can get, what challenges we might have, and how we can overcome this. So the primary aim of this project was to assess the feasibility of wearable fNIRS to be used on freely moving toddlers. And also as a second aim, we also wanted to validate a virtuality platform to assess the development of executive function in toddlers. And this is what we've just presented at the poster session. So in terms of data collection, this is currently ongoing. So we are recording data on both adults and children. So far we got 5 children plus 3 just before I left for Boston. And we are recording both fNIRS data combined with some other recordings. So we're using a dual bright system from Martinis with 48 measurement channels, 4 of which are short separation channels. So we are recording data from dorsolateral PFC bilaterally and also motor cortex to do some sanity checks on the data as well. So basically participants are standing in our cave lab, which you can see in the, does it work? No, in the cave on the top, in the picture on the top right. They're wearing the NIRS cap, they're wearing shutter glasses for stereoscopy. They're also tracking the movement of the head and of the end, so they can, the space can rotate with them, and also they're able to interact with some objects. And of course, the NIRS cap and the backpack. Okay, in terms of the experimental protocol, so we wanted to start with something quite simple where we will know more or less what type of patterns of brain activity we would expect. So we use block design go-no-go task, and all participants completed a virtual reality version of the task and a computer-based task similar to the one that we are typically using in lab settings. So the task included Go blocks with Go-only trials and mixed blocks with Go and no-Go trials. So in the VR version of the task, basically participants are in a virtual playground, and there are bubbles of 2 different colors coming up in the space. So during the Go-only blocks, participants are asked to pop only the blue bubbles. And in the mixed blocks, yeah, they're asked basically they don't need to respond to the red bubbles. So we want them only to pop the blue bubbles. And the bubble popping is achieved by using colliders linked to the motion tracking of the hand. In the computer-based version of the task, instead, they're doing a classical computer-based experiment where we're using button press basically to trap the bats, but not the cats. In terms of data analysis, we started off with a pretty standard and simple analysis pipeline that involved motion artifact correction, bandpass filtering, and block averaging across the Go and mixed blocks conditions for both tasks. So today I'm going to be focusing mostly on the VR, some data from the VR task. So first of all, we started to look a little bit at the data quality that we could get out of 3D moving participants. So here I'm showing one channel for 4 of the participants that we recorded so far. These are the raw intensity data, the 2 wavelengths with no preprocessing applied. So on the top, on the example on the top, so for participant 1 and 2, I will say that we're getting pretty good quality data. So we don't see any major motion artifacts. We do see some probably, hopefully, task-related modulations in the signals, on top of which we see a clear heartbeat component. In the 2 examples on the bottom instead, we do observe a little bit more motion, but most of the artifacts that we are observing both on the adults and on the children are fast spikes rather than very big, large baseline shifts, which is kind of a good news for us because these types of artifacts are a little bit easier to correct than baseline shifts. If we look now on some examples of concentration data, here I'm showing some time series from channels over our region of interest, that is dorsolateral PFC, across the whole duration of the experiment. So these are preprocessed oxyhemoglobin signals in red and the oxyhemoglobin signals in blue. And it's very nice to see that we observe quite nice and consistent changes in brain hemodynamics, which are phase locked to the presentation of the stimuli. So we see increases in oxy and decreases in deoxy across the Go blocks and the mixed blocks. So if we now try to average our signals across the 2 task conditions, we can look at some mean hemodynamic responses across a few channels. So here I'm showing the block average hemodynamic responses from one of the participants. Red is oxy, blue is deoxy, and the mean hemodynamic response to the mixed blocks is shown in solid line and to the Go-only blocks in dashed line. So looking at the data over our region of interest, so dorsolateral PFC, it seems that we are observing the expected parts of activity, so larger changes in brain hemodynamics to the mixed blocks compared to the Go-only blocks. And we are now we're looking also at some data over the motor cortex to assess if we are getting something meaningful and robust. So participants basically are popping the bubble, moving the right arm. So what we are finding is that there are larger changes in brain activity in the left hemisphere. And the level of activity is quite comparable and similar across both the Go blocks and the mixed blocks, which kind of makes sense because the same amount of motor activity is required for both conditions. So this suggests that we might be getting some meaningful and robust data. So hopefully it is not totally random. So in terms of some preliminary discussions based on our experience so far, we're finding that, of course, the VR task is a lot more engaging for the children. So with the computer-based task, very often children get quite bored, they want to stop the experiment, and this is not happening with the VR task. So all the participants so far, so 8 in total, completed all the blocks, and we didn't have to exclude any trials due to poor attention or looking time. In terms of equipment, so far, none of the children have complained about all the headgear and all the equipment that they were wearing. So we think that this might be because they are quite engaged with the task, they feel immersed in the space, it's kind of fun for them that they probably forget about everything that they're wearing. And when working with these populations, we also realized that we can also take some time at the beginning of the experiment to optimize the optical coupling and play a little bit with the cap. Also, thanks to, we are taking advantage of the cinema size screens of the cave. So we project Paw Patrol, the kids are sitting in front of the cave, and we can take all the time that we want to play with the cap. Lastly, so far, I will say that these preliminary observations, that the data that we're getting are a pretty good quality, and as we are measuring the expected patterns of hemodynamic activity. But of course, we're getting more larger amount of motion artifacts in the VR data compared to the computer-based data. In fact, this is just a summary. So here I'm showing the number of detected motion artifacts using the OMER2 routine across the participants that we've tested so far. And of course, we have a larger amount in the VR task. But overall, it seems that with the available algorithms, this can be corrected properly, and we can recover quite a robust and nice hemodynamic response from this population. So after these preliminary and general observation in the data, we started to dig a little bit more into the signals and try to understand what other type of noise we can get in this situation. So participants that can freely move and they stand in the environment. So we all know very well that the NIRS signals that we are measuring are actually a mixture of components of both neuronal and systemic origin that originate both at the cerebral levels, but also the extracerebral compartments of the head. And you can well imagine that in our case, we have a lot more movement, a lot more posture changes, as also Alex von Luhmann has shown yesterday. So this means that we get, probably very likely that we get larger changes in systemic physiology, as well as some of the others noise components in the data. And also previous work has shown that myowaves, so the 0.1 Hz modulations related to blood pressure regulations, have actually a larger impact and a larger interference when NIRS data are recorded on participants who are standing compared to when they're sitting, compared to when people are laying down. So in our case, we have participants who are standing and they're moving a lot more. So one possible way to account for some of these scalp changes and physiological noise is to use short separation channels. So putting a source and a detector close to each other to sample the data from the scalp, which can be regressed out from the long separation channels. In previous work, has actually demonstrated that by regressing out the short separation channels from the long separation channels improve the recovery of the hemodynamic response, especially when the myowaves, so the 0.1 Hz component, have quite a strong amplitude in the signals. And also the performance of the regressions improves if we combine superficial regression also with other additional nuisance regressors like accelerometers. And this is what Alex has presented yesterday as well. However, all of this has been investigated quite a lot in adults, but not in developmental populations, except, to the best of my knowledge, only the work by the great Lauren Emberson a few years ago have actually investigated whether scalp changes occur in infants as well. And what they found is that, yes, there are changes also happening at the extracerebral layers of the infant's head, but when we regress out this information from the long separation channels, this does not really have an effect on the group level statistics. So here we wanted to understand where toddlers stand. So are we in a situation that is more probably similar to the adults, or probably it's not useful to have this additional information. So as I said before, in our array, we had 4 short separation channels. So 2 on the forehead and 2 more towards the back, towards the motor cortex. So So if we look a little bit at the data from some examples from a long and a short separation channels for one of the toddlers, which I will call toddler number 1. This was 3 years old. So these are raw intensity signals with no preprocessing. So if we look at this data either in the time domain or in the frequency domain, we couldn't really see any strong, maybe Maya waves components standing out from the data. So the data look, there is some physiological components, but not a strong visible Maya wave, which was what we were actually observing in almost the 90% of the adults data sets that we have recorded up until now. But if you look at another toddler, so this is what's called toddler number 2, and this one was a 5 years old kid. Here already visually, just by looking at the time series of the data, we do see quite a clear and strong 10 seconds components, which are related to, these are the Maya waves component basically. And already while we were recording the data, we could see already visually in the real time during the acquisition that these were quite strong and stood out from our signals. So to understand a little bit more, we started to look at these signals a little bit more in depth. So here I'm showing the mean hemodynamic responses recovered from the short separation channels for toddler number 1 on the right and toddler number 2 here on the left for the VR task on the top and the corresponding channels in the computer-based task. So if we look at data from toddler number 1, who was the one with not very clear physiological noise, we don't really have very strong changes happening in the superficial layers of the head, so we get quite small hemodynamic changes. But if we look at the data from toddler number 2, and particularly in the VR task, so when the kid was actually standing and moving around, we get quite large changes of about 0.6 micromolar, especially in the oxygenated hemoglobin. And it's also very interesting to notice that the 10 seconds component actually survives the averaging and is quite clear. This is not the case of the computer-based task. So when the same kid was actually sitting down, we don't really get such a large change in hemodynamics. So to understand a little bit more how the short separation channels regression impacts the recovery of the hemodynamic response, we applied 2 separate pipelines. So in pipeline number 1, we did not perform the superficial regression, while in pipeline number 2, we did correct for short separation channels. So we regressed them out from the long separation channels. So if we look now at the comparison, the results, if we apply pipeline 1 and pipeline 2, so here I'm showing the block average hemodynamic responses from the 2 toddlers from a few channels. So these are all long separation channels. And red is always oxy, blue is deoxy. Results by applying pipeline 1 are shown in a dashed line, and results for pipeline 2 in solid line. So if we look at the toddler number 1 here on the left, in either the VR task or the computer-based task, it seems that superficial regression doesn't really change the situation much. So it doesn't really have a strong, the correction is not that strong. But in toddler number 2, so the one where we get very strong components, for the VR task, the correction seems to be quite significant and quite strong, especially on the oxygenated hemoglobin signal, as you would expect, as it is more prone to systemic interferences. And here, for example, in one of the channels on the top right, we can see that the amplitude of the response has been attenuated, and also the 10 seconds component also has been kind of interpolated and reduced a little bit. Looking at the computer-based tasks, so when children are actually sitting down, we are in a similar situation as toddler number 1, and the regression does not really change our signals significantly. So before jumping off to the conclusions, I just wanted to make a quick note on multimodal neuroimaging with NIRS on children. So previous work has actually demonstrated that it can be highly beneficial to measure other signals alongside fNIRS, especially in situations like naturalistic experiments. And these can be monitors of behavior, monitors of physiology, so we can do what Felix Scholtmann calls systemic physiology augmented fNIRS, or coming from other brain modalities. And having this additional information can be useful for various reasons. So first of all, they can help us in interpret the patterns of brain activity that we get. can also help to explain the variance in the fNIRS signal, so we might be able to get a better denoising and get rid of some of the noise components that cannot be removed by a simple bandpass filter. And lastly, they also provide complementary information about cognition, brain functioning, and physiology. So if we want to do something like this on children, we need to consider various challenges. So first of all, there are not many off-the-shelf, child-size, integrated, wearable, multimodal devices that are integrated with fNIRS. So for example, in our CAVE lab, we found ourselves to have to integrate fNIRS with motion tracking, wearable height tracking, and shutter lenses for stereoscopy. So we want to get in a situation like this. So what I had to do is I had to design and 3D print a custom glasses frame that could accommodate all these sensors, which also had to be quite lightweight and also easy to be put on top of the NIRS cap, and of course able to give us robust data. Second, with children, we might also want to think about what are the parameters that are more informative for our particular experiment. So of course, we cannot put lots of equipment on the children. It will get quite uncomfortable and quite difficult to set it up. So we might need to make a selection of what is more important. Third, a very big issue is synchronization. So since we need to create our own multimodal platform with wearables, we need to also ensure that all the data streams are synchronized. And at the moment, I'm handling this using network protocols, like lab streaming layer, which is working quite well. And lastly, some of the monitors that I mentioned before, like eye tracking or motion tracking, also use infrared radiation, which can interfere with our fNIRS measurements. So for instance, we were just about to start mockups of motion tracking fNIRS experiment, and we've noticed that since the motion tracking cameras shine infrared light at 810 nanometers, we got a lot of interference in our NIRS signals. So we started by characterizing a little bit this interference to understand how can we block these interference and get clean NIRS data. So Musa Talati has just presented his work at the poster session, but basically we recorded some NIRS data from various regions on the head in the preschool lab where we have 15 motion tracking cameras. So here the amount of external infrared radiation was quite a lot. And we were recording motion tracking at 120 Hertz and fNIRS signals at 25 Hertz. So our acquisition protocol consisted of 2 minutes of recording with the cameras off and 2 minutes of recording with the cameras on. So if we look now at some examples of NIRS data that are actually impacted by the interference, here I'm showing the raw intensity time signals, time series from one of the channels. Here was from the frontal cortex, shown in red. If we look at the first portion of the signal, so the first 2 minutes with the cameras off, we don't really see anything weird.
1 minute.
Okay. We don't really see anything weird in our data. We see the usual NIRS signals with the heartbeat rate. But if you look at the second portion of the signals, we already see visually that there is something really odd in our data. And the interference was actually some very high frequency noise, higher than the heartbeat rate. So if we look now at the same signals in the frequency domain, with the cameras off on the top and the cameras on the bottom. In both cases, we see like a nice, clear heartbeat component with its harmonics, but when the camera's on, we do see a quite sharp and strong component of 5 Hertz that in our case corresponding to the aliasing frequency, since the motion tracking cameras are recording a much higher sampling frequency than NIRS. So we started to, so this is for motion tracking, but actually in this case, we could probably just use a low pass filter to get rid of this noise frequency. But there are other cases like with some eye trackers where the interference might be really difficult to sort out with signal processing and might impact our NIRS signals at all the frequency, especially the hemodynamic component bandwidth. So we start to discuss a little bit how can we block these interference. We were discussing with Elias possible solutions. So one of these was actually to put like a cover up on top of the NIRS cap to block this interference. But we want this material to be quite cheap, easy to find, not too thick, and easy to be put on children's head. So Elias has an idea to actually use a tin foil and not only to block our minds for the aliens. So what I've done is I've sewn like a cover up for the NIRS cap that was made of 2 layers of dark lightweight fabric with a layer of tin foil inside. And we took again the same measurements, so 2 minutes cameras off, 2 minutes cameras on. And again, in this case, we are not in the same situation as we were before, so we don't really have anything odd happening when the cameras are actually switched on. Looking at the signals, again, in the frequency domain, we still see the heartbeat rate, but we don't see the 5 Hertz component anymore. So it seems that this method works quite well with our data set and might be possibly a useful addition when using external monitor that are based on infrared radiation. Right, so to wrap this up, based on these preliminary investigations, it seems that wearable fNIRS is being well tolerated by toddlers and preschoolers, and we can get pretty good quality data considering the context in which these are recorded in. Of course, we need to take a few aspects into account, which one of these is systemic changes happening when participants are freely moving. So far we're finding both in the adults and in the children that there are higher scalp changes when in the VR task, so when people are standing up compared to the computer-based task. And also these components are a lot more variable across children than adults. So in the adults, it was pretty consistent, around 90% of our data set shown the same pattern. But with children, it seems that these become more consistent on older children, so on the 5 years old compared to the 3 years old. But of course, we need a much larger sample size to make this conclusion. So far, we actually recommend that we're recording data on toddlers, on these younger, on these children. We recommend to include short separation channels in our array. Finally, when using infrared-based devices, we also recommend to characterize the source of interference and take appropriate measures, either signal processing or something more practical during data collection. So the next steps are, of course, to complete data collection and also to combine short separation channels with other signals. For example, we are recording acceleration that is integrated within the NIRS device, but also head movements. So we're planning to apply TCCA on this data. I would like to thank Alex Luman for the useful discussion about this. And lastly, we will start to test the feasibility of systemic physiology augmented fNIRS on children, understand better the link between brain and physiology, and whether this information can help us to get even better data. So this is the end of my talk. I would like to thank you for your attention. And of course, special thanks to our young scientists, the UCL group, with which I've embarked in the journey of the real-world neuroimaging a few years ago, and my new group at the Total Lab Birkbeck. Thank you.
Very excellent talk, a lot of fun. We have a couple of minutes for questions. While you're coming up, do you have a sense of how much all of that gear weighs for the kids to be wearing?
So it is approximately 400 grams, the NIRS cap, and the glasses are less than 100 grams with all the key panel lenses and markers, etc. And
then how long can the imaging session last?
So far, the experiment lasts like 10 minutes for each task, so like 20-25 minutes for both experiments, but they did not complain about the cap, so probably we can get even longer.
Yeah.
So 2 questions. The first one, have you done baseline comparisons to just being like standing and moving and not just doing VR tasks? Because I guess just to see, is there any difference between just natural increase in blood flow because you're doing activities in the motor cortex and the cerebral area for balance? And then the second question is kind of related in the sense you haven't used a lot of kids yet, but some people have motion sickness due to VR. Does that cause any background noise, or have you noticed anything or account for that?
Yeah, I'll answer the second question first. So I think that the cave is a little bit different from the VR headsets that we usually wear. So in the cave, you have a much better sense of your body. You feel your body in the space. you have more control, you get less motion sickness. But of course, with the children, we design the tasks that are child-friendly. So there are not like rapid movements in the environment or anything that would cause them to feel sick or anything. But so far, none of them have reported issues with this. Actually, they call the glasses magic glasses because they can see everything in 3D, and they are pretty happy to wear that. And your first question was about baseline comparison between the VR and the computer-based task. We haven't done that yet, but our comparison is mixed blocks versus Go that basically should include the same amount of movement and systemic physiology. So ideally, we are kind of subtracting this effect out a little bit also when comparing our conditions.
Awesome. Thanks.
Okay, in the interest of time, we're going to move on. So thank you very much, Paula, for the wonderful presentation.
Good afternoon, everyone. It is always a pleasure to present and introduce Professor Fantini. He's today going to talk on the coherent hemodynamic and dual slopes for non-invasive optical measurements of the human brain. Professor Fantini, thank you very much.
Thank you very much. Thank you very much, Felipe, for the introduction. And I'll be glad to talk about a couple of activities that we have been developing in my lab here at Tufts University, close by. And those are coherent hemodynamic spectroscopy and dual slope methods for non-invasive optical measurements of the human brain. So one of the strengths of fNIRS is the fact that it is based on relative measurements over time. And coherent hemodynamic spectroscopy is the same. It measures oscillations or transients in hemodynamics that allow for studies in the time frequency space. So this is something I will not be talking about today, but another feature of CHS is to allow for quantitatively relate the measurements of oxygen deoxyhemoglobin concentrations that we measured in fNIRS with underlying physiological changes in blood flow, blood volume, and metabolic rate of oxygen. One fNIRS limitation, which of course is well known in the field, is the sensitivity, sometimes the strong sensitivity to superficial extracerebral tissue. We have seen Paula was talking about the short separation channel and aggressing out the signal out of the long separation channels. Here we talk about dual slope methods that use only long distance measurements to achieve this selective sensitivity to the brain. So the first measurements I want to show is of cerebral hemodynamics that are coherent with physiological arterial blood pressure changes. And in this protocol, we used two methods to induce oscillations of arterial blood pressure. One is a thigh cuff that is inflated on the thighs of the subject, and the other one is pace breathing by asking the subject to follow a breathing metronome. We did measurements in this case at multiple source detector distances. That is the probe on the right. A is a single detector, and 1 through 7 are light illumination points that achieve measurements at distances between 11 and 40 millimeters so that we can perform single distance measurements, SD, single distance ranging, of course, from 11 to 40 millimeters, or what we call single slope measurements. So slope measurements that are based on a single element, in this case, a single detector A, and then we can group the illumination points 1 to 3, 2 to 4, 3 to 5, and so on to achieve slope measurements at varying source detector separation. And this is essentially the top panel as a function of time is the protocol, the cyclic cuff inflation period and the pace breathing period. And then the second plot is arterial blood pressure. And so you can see the oscillations induced by cyclic cuff inflation and by pace breathing. And then the bottom three panels, red is oxy, blue is deoxyhemoglobin, represents the coherent cerebral hemodynamic oscillations that happen during the periods of cyclic cuff inflation and pace breathing. And that is the emphasis of what I'm showing. And really I would say there are two slides in this talk that are crucial. And this is the first of the two. And then if you tell me my time is over, after the second, that's fine. Now I will just go to the conclusions. So here you see, as a function of source detector separation from 10 to 40 millimeters, the top panel is the amplitude ratio of deoxy versus oxyhemoglobin oscillations at 0.1 Hertz, which is the frequency at which we induce the driving blood pressure oscillations. The bottom panel is the phase difference between the deoxy and oxyhemoglobin oscillations. The first message is very clear. Psy-CAF oscillations and pace breathing essentially give the same results. One is dashed line, the other is continuous line. So the point is that it doesn't matter how the blood pressure oscillations are achieved, their impact on cerebral hemodynamics is essentially the same. The second point is that there is a transition from superficial hemodynamics and deep hemodynamics. So when you go from source detector separations of 10 millimeters to 40 millimeters, in the amplitude ratio of deoxy versus oxy increases, and the phase difference, bottom panel, transitions from about in phase, that would be minus 360 degrees, to about in opposition of phase, that would be about minus 180 degrees. So somehow the nature of the tissue hemodynamics driven by this systemic blood pressure is different. The nature is different. On the scalp, on the surface, you have in-phase oscillations with low ratio between deoxy and oxy amplitudes. And as you go to brain, you have out-of-phase oscillations with a larger amplitude. The third point is that if you look at phase measurements, those are the green lines, even though they're single distance measurements, they already, even at short distances, they're already representative of the deeper hemodynamics. See, larger D overall ratio and opposition of phase oscillations. So somehow we have an indication here, which also is well known in the field, that phase measurements probe deeper to the tissue than intensity measurements. So let's use a phasor representation. We're dealing with oscillations at a given frequency, so we can use phasors, which are vectors in the complex space, where amplitude is the amplitude of the oscillation and phase is the phase of the oscillations. So if we have CBV, cerebral blood volume oscillations, oxy and deoxyhemoglobin will oscillate in phase with that, and the ratio between the amplitude of oxy and deoxy is a measure of the hemoglobin saturation of the oscillating compartment. Of course, we also have blood flow oscillations, and in general, blood flow and blood volume are not going to be in phase with each other. But here, for the sake of argument, let's say that they are in phase. So one thing that's coherent hemodynamic spectroscopy and the model associated with that found is that the associated oxyhemoglobin concentration changes are out of phase, they're lagging the driving cerebral blood flow oscillations. And the reason for this time lag is because of the transit time of blood in the microvasculature. And then considering here two cases. On the left, it's a case where blood flow and blood volume contributions are comparable to our oxyhemoglobin measurements. And on the right, instead, blood volume dominates the signal. So let's focus on the case where blood flow and blood volume are about the same. If those are the contributions from blood volume and blood flow to oxyhemoglobin, the red phasors, the big red line is what we measure. It's just a vector combination of the blood volume and blood flow contribution. And on the top, there is deoxyhemoglobin. And then the phase angle between them is about minus 200 degrees. And now we can plot the phasor ratio, which is a vector, D over rho, which is the amplitude ratio of deoxy versus oxyhemoglobin, and the phase. If you do the same thing on the right, where blood volume dominates, we see that those are the measured minus 330 degrees. Now we're getting close to being in phase, and that's what we get. And you recognize here that on the right is what we measure on the surface, on the scalp, and on the left is closer to what we measure in the brain, where the rigid enclosure of the skull limits the blood volume contribution, and you have a more relevant blood flow contribution. So that's where coherent hemodynamic spectroscopy allows for any interpretation of these results. And also, this stresses again one point that this community has been making for years of the relevance of always reporting both oxyhemoglobin and deoxyhemoglobin results in fNIRS studies, because they really provide a more complete picture of the physiological changes behind the fNIRS signals. So the next topic is the dual slope, which is an extension of single slopes. So single slopes are based on a single element, let's say a single source, source one, and two detectors. So one can measure a slope But then, of course, one is sensitive to any kind of contact or detector sensitivity contributions. So let's say the detector 1, which is a short distance here, has an added contribution because of its response function. If we now consider a symmetrical single slope based on source 2, now you see what happens. Detector 1 is now the long distance. So if you have an added contribution from detector 2 at the long distance, now the slope that you measure is steeper, while for single slope 1 is shallower. It turns out you just take the average of the two and you correct for any coupling with the tissue, any detector sensitivity response, any source emission properties. And so this is a very powerful feature. And I have to say, it's a brilliant idea. And I can say that because it's not my idea. It's Dennis Huber of ISS. He had it in the late 1990s. I said this several times when I present this. But I believe that this is the only time that Dennis Huber is indeed in the audience. So I really want to give this recognition to this idea. And this idea has been used by several groups in this community. And I know of at least 2 companies that have implemented them in their probe, as shown here. But there is one thing, and that is what we have been focusing on over the past few years, and that is the regional sensitivity associated with dual slopes. We know that for a single source detector pair, we have this banana regional sensitivity, intensity in the top, phase in the bottom. It's well known that the phase regional sensitivity is deeper. But when you have this dual slope configuration, something different happens. So you have a regional sensitivity that is now more localized and is deeper. So we say that those are more like nut regional sensitivities as opposed to banana regional sensitivities. Recognitions to Giles and Angelo in the group who have been doing a lot of work in this area. So the point is that with this dual slope, in addition to achieving this self-calibration and insensitivities to instrumental contributions, to coupling artifacts, to motion artifacts, you also achieve this preferential sensitivity to deeper tissue in the brain, and intrinsically less sensitivity to the scalp and skull. So I should also report that Adam Liebert, at the same time and completely independently, came up essentially with the same approach. Here again, you see the 2 sources that sandwich the 2 detectors. And in the center panel, you see the first moment of the time of flight distribution, which is essentially the phase in the frequency domain. On the top, the banana sensitivity associated with single source detector. On the bottom, the nut shape associated with this dual slope configuration.
3 minutes.
3, 2, 3, 3. 3 is plenty. So these are the measurements we did with this dual slope configuration. Essentially, it's the same thing. Now we induce blood pressure oscillations with the cuff. And again, red and blue are the coherent hemodynamics now measured with single distance and single slope. And there it is. This is the second key slide. So as long as I go through this, I'm happy. This is what we measure in terms of the phasor ratio. So these are vectors of D over O. Those vectors, the amplitude represent the amplitude ratio of deoxy versus oxyhemoglobin oscillations. The phase represents the phase difference between deoxy and oxyhemoglobin. And now you see that at the single distance, short, 25 millimeter, which is still long in the sense that it is sensitive to the brain, and the 30 millimeters, you see these small deoxy to oxy ratios and this phase that is closer to 0 than to 180 degrees. Then the information content about the brain, of course, is there, but it's overwhelmed by the superficial hemodynamics. You combine those 2 pieces of information into the dual slope vector, which is that orange or yellow, and now you have the much larger deoxy over oxy ratio and a closer phase to 180 degrees. So we have a dominance to blood flow rather than blood volume, as it is the case for the scalp. And then when you go to phase measurements, you see that even the single distance measurements at 25 and 30 millimeters, now they already have a stronger representation of the blood flow dominated cerebral hemodynamics. And the dual slope further enhances that. So this is the key slide. And then the last thing I want to show is how to implement this into imaging of the brain. This is a dual slope imaging probe. There is a collection of sources and detectors that feature a number of dual slope units. They're placed on the occipital region for the standard visual test. And here, all I want to show, the protocol is well known. I want to show the results. And these are, at the end, this is what the goal is, to have more sensitivity to the brain. On the left, there is a single distance intensity measurement with a typical hemodynamic response to brain activation. The dual slope arrangement already enhances the sensitivity. The hemodynamic response is larger. And when you go to phase measurements in the frequency domain, single distance is the same as dual slope. And as you go to dual slope phase measurements, you have an even larger sensitivity. So conclusions, I talked about coherent hemodynamic spectroscopy and dual slope. And I guess the point is that phase measurements are more sensitive to the brain than intensity measurements, and dual slope measurements are more intensive to the brain than single distance or single slope measurements. And finally, many thanks to all of the lab members and to the funding agencies. Thank you for your attention.
Very beautiful talk. Very cool stuff. We have time for just a couple of questions. Mari?
Great talk, Sergio. Nice to see you here. We have the system of fNIRS that is based on this dual slope, and we observe some problem when we have some inhomogeneity under one part of the combinations. So like if we have a big blood vessel, superficial or something, things get very messed up, and we see in some cases even the opposite. Do you have simulation with the inhomogeneity, and how do you handle those cases?
Yeah, thank you. That is true. And actually, we have done lots of simulations of inhomogeneous tissues. And indeed, they create problems, especially for recovering of absolute properties. For recovery of changes, hemodynamic changes in the brain, they usually compensate those. But it is true that the relative behavior of phase and intensity may be different. Also, the fact that you have a top layer that is more scattering than the bottom layer is at more of a disadvantage for phase measurements and intensity measurement. So it is true. Inhomogeneities are an issue to be addressed.
Thank you.
All right, Stefan, one more question.
Okay, quick question. I was wondering, so the sensitivity of phase is higher. Did you do any evaluation, either simulation or experimental, of contrast to noise?
Yes, that's another very good question. And in fact, we tend to speak more about contrast to noise than signal to noise or sensitivity. Clearly, phase measurements do suffer in terms of noise, but we have observed that once your measurement allows you to get a sufficient contrast to noise, then you're always going to be beneficial. But it is true, there are cases, we have seen cases where we could not have enough contrast to noise in phase measurements, and so we had to rely on dual slope intensity.
Thank you.
Yeah, it's another issue.
Sergio, following from that, we've seen similar result that phase measurements have worse SNR, but they do give more contrast in the recovered parameter. And what we found is combining intensity and phase together gives you the best of both worlds. Have you tried that?
So we haven't tried combining them for the goal that we're after, which is that of achieving more sensitivity to the brain. But as I was saying before, we have cases in vivo where we just could not achieve enough contrast to noise ratio in phase measurements. So I guess the brief answer is no, we haven't tried it, and definitely it is worth trying that. But let's say our approach has always been, if we can get those dual slope phase, let's go after those.
Right. So let's thank Sergio again. Wonderful talk. Thank you so much. So our next speaker is Sarah Sanchez Alonso. She's going to be speaking about the correspondence between simultaneously collected fMRI and image reconstructed fNIRS using both individual anatomy and a structured light 3D scanner.
Okay, great. So how do I move to my side? Well, first of all, I want to apologize for the slides. I don't know what I'm going to find because I was notified this morning that I had to send a PowerPoint instead of Keynote. So I know they've done a transformation. So we'll see. Oh, it's a PDF. Oh, that's good. Oh, yeah. Oh, it's a PDF. Okay. Should I do it here? It's in a good place. Okay. All right. So it's a PDF. Is it that? I don't see it there, right? I mean, it's here. Yes.
One moment, please.
Oh, maybe this here, there. I mean, it should be coming up. Yeah, right.
It's not the right talk.
No, it is the right talk. It's just not. We don't see it
Oh, no.
on the screen.
Slides?
Ah, okay, perfect. All right. Well, if this works, then I think at least we'll be able to see my slides. Ah, okay.
Okay.
All right. So, all right. So it's a PDF. So I think we'll be able to see my slides at least. Okay, great. All right. So this actually works pretty well. Right, so today I'm going to be presenting data from a study in which we are studying the correspondence between fMRI and NIRS signals collected simultaneously. First, I want to say from the start that this is very much a collaborative work that has been conducted in collaboration with an amazing team of scientists that you can see here. I also want to acknowledge the funding sources and SfNIRS, who has provided the system that we are using, and they've been very supportive throughout the whole study. Like I'm using this. First of all, I want to think about the motivation, right? We are not the first ones to do fMRI, data collection of fMRI and signals simultaneously. There have been several groups in the last couple of decades. Many of them are here today in this room. So I want to start thinking about what are we contributing to what has been done so far. And one of the reasons why we have started doing this study is because we identified several gaps in the literature that we still don't quite, that we still need to fill in. And one of our goals was to understand neural and behavioral variation across development. And this is challenging, right? So although fMRI is the gold standard methodology in adults, it is difficult to implement in with infants and children. Plus, we know that the environment is very important for learning, right? So capturing that environment and having that environment there is critical for us. So in the current study, what we are doing is conducting a quantitative comparison of fMRI and NIRS signals conducted simultaneously. We are using crucially a full head coverage of the NIRS array, and as I mentioned, simultaneous data collection, and we are doing this both in adults and in children. So the core question that we're trying to answer is, to what extent are the two signals comparable across development? I'm going to focus today on the study with adults that we've been conducting. So here's an overview of some of the key features of the study, just so that you can see how we are building in prior literature. So in addition to collecting both the data simultaneously, we are using a full head NIRS array, simultaneous measurement of physiological signals along with short channel measurements. We are using two co-registration methods for mapping the optodes to the brain tissue. I'm going to talk a little bit about that in a moment. And we are also assessing the test-retest reliability of the signal across 2 different sessions. And finally, we are including developmental populations, which is one of the populations that hadn't been studied in the past. So I'm going to walk you through the study setup. This is the device that we are using is a NIR Scout X. Here you can see a picture of the cups. As you can see, if you're familiar with the NIR X systems, you can see that the optos that we are using are flat. We actually really like the system. You basically clip the optos on the cup and they don't move. We've made quite a lot of improvements on the cup. Among them, one of them has been to glue the grommets to the cup, so that it doesn't move at all while we are setting up the participant and then lighting the scanner.
Here you can see the optical fibers. They are 10 meters long, and you can see them here in the control room. We bring them through the waveguides into the scanner room. Here you can see a picture. We have to build this bridge in order to bring the fibers into the scanner table. And here you can see a picture of one of the participants in the scanner. As I mentioned, we are measuring simultaneous data collection of physiological signals, in particular heart rate and respiration. With respect to the array, here's a picture. You can see that we are using short channel measurements in green. The distance is of 8 millimeters between sources and detectors. And you can see a picture as well of the short channels. I also mentioned earlier that we are using two methods for scout to anatomical registration. So one of them is we are using vitamin E capsules as fiducia. So basically we put a vitamin E capsule next to every optor on the NIRS cap that allow us to identify on the T1 image the location, the exact location of the optor. We are also using a 3D structure sensor. This is the reason for this is because this is the system that we use outside of the scanner. So it was very important for us to have that comparison. If you're not familiar with the scanner, I don't think the video is going to work, but here is a picture of the scanner. You just basically put it on the laptop, and the way it works is it projects a known pattern, in this case, the head, onto a scene, and the way this pattern deforms when striking the surfaces allows the scanner to calculate the depth and surface information. I won't be talking more about this system today, but if you're interested in the protocol that we are following, I'd be happy to talk more in the question period of after the talk. So I'm going to move on now to the experimental design and just show you the data that some of the preliminary data that we've been analyzing. But I do want to mention that the study setup, just setting up the study, took us about 6 months. So there are a lot of details and things that we have to figure out in the process that I'm glossing over here. We are actually publishing the protocol with a description of the exact setup that we followed. It's going to be published in the Journal of Visual Experiments in a couple of months. So stay tuned for that if you're interested. So let's go on to the experimental design. So we've collected data from 40 young adults, all of them native speakers of American English. They come for 2 sessions. In one of the sessions, they watch 2 movies for a total of 15 minutes, and they also do 2 rest and stay sessions for a total of 10 minutes. They come back for a second session, and here they do again 2 rest and stay sessions for a total of 10 minutes. This is for us to test the test reliability of the signal. And we are also doing a traditional language localizer that has been used in larger scale data sets such as the Human Connectome project. Is the reason why we included this task, and also a visual task, a flashing checkerboard task of 6 minutes. So just so that you get an idea of the involvement of the project, each single session takes about 2.5 hours. The participant is in this scanner for about 35 minutes, but it's just the setup that and then the cleanup that requires quite a lot of time. Here's just an overview of the methods that we follow for the data processing and analysis. You can see our software tools to the left. We use QNX for analysis of the fMRI data and NeuroDot for analysis of the NIRS data. I just want to highlight a couple of things here in the interest of time. For the fMRI data analysis, we use a standard Human Connectome project processing pipelines. For analysis of the NIRS data, we are doing image reconstruction and also light level time series analysis, of course. And then we are bringing most of the signals to the surface and doing task evoke analysis of the 2 data. There is actually there's an edit that has to be done to the graph. Actually, for motion artifact correction, we are actually using the global variance of temporal derivatives. It's kind of like a similar measure that is used in fMRI as well to assess motion. All right, so let me now show you a little bit of the data that we've been collecting. So here's Here's a picture of the sensitivity profile of the NIRS array. So basically what we do is that we model for photon transport through the brain tissue. And what you see here is the sensitivity matrix, which basically allow us to predict based on how optical density measurements change through brain tissue, allow us to predict how much data a single measurement is able to collect based on the location it is on the brain. Here's just, I'm going to show data from one of the subjects. So remember that these are young adults. They don't move much. So we have very minimal movement in this data set. As you can see, the mean light levels are very good. We lose a little bit of SNR on the top of the head. This is as expected, especially with some subjects that have smaller or larger heads. The areas, the cup doesn't have that good of a contact in sensory motor areas. It doesn't happen in every one of them, but it happens in some subjects. And with respect to measurements, here's a representative subject. We only lost a single channel on the next to sensory motor areas. Here's the raw data with all the channels. You can see at the top all measurements by wavelength. And in the bottom arrow, you can see is the similar picture, is just thresholded by standard deviation. Now I'm going to show you the pre-processed data. So on the top, you can see the raw data after pre-processing, after bandpass filtering.
2 minutes.
Thank you. And below you can see the Fourier transform data, which allow us to see the frequency components, and we can see the peak at the stimulus frequency as expected. And here is the task. I'm just going to give you more details about the visual. Sorry, so I'm going to be presenting data only about the flash and checkerboard task. So we have 10 seconds of flash and checkerboard. We have 20 seconds of rest, so on and so forth until for 6 minutes. Crucially, at the beginning of the resting state period, there is an attention check in which the participant has to press a button to turn the circle red. And as you will see, there's going to be some engagement of motor areas expected at the beginning of the rest. Here is the result of the block average data for this subject. As I said, this is very clean data, so nothing is looking as expected, with strong bilateral activations in visual cortex areas. Here is the whole the block average data by a slide. So as you can see, we can see already the activation. We can see very well here the activations in sensory motor areas as well, close during the rest period. So they're reflected here, which is very nice. And now I'm going to move on to the fMRI data. So here are 3 participants. Here, the signal is as expected. We are using the 20 channel head coil instead of the 64. So we were not sure whether the data would be as expected or not, or if we would see any differences. But we get clear bilateral activations in V1 and V2. And if we zoom in into this subject 1, we can also see that the LGN is engaged as expected. So at this point, you may be asking me, well, do you know something about the correspondence between the 2 signals? And my answer is that not yet, because we are missing a crucial step that we are working on right now very actively. And this is, we are working on an algorithm to automatically identify the fiducials on the T1 in order to extract the opto coordinates. And we are also doing tissue segmentation of the T1 images. So our next step is to use this information to create the light model and analyze the data and be able to actually look at the correspondence with a special precision. So I'm just going to give you a summary. So I've told you about our fMRI, simultaneous fMRI study and how we built on prior literature, particularly using full head coverage, short channels. Our current NIRS pipelines include image reconstruction. This is crucial in order to measure photon transport through the brain tissue. And in order to, once we have the segmented brain tissues specific to a subject, be able to use those images to identify the exact location of the optoids. model the photo transport more precisely. The data that we've been analyzing looks very good. It's very clean data. We see a strong engagement of V1 and V2 in the fMRI data and visual cortex areas in the NIRS data as expected. And as I mentioned, our ongoing analysis are focused on achieving a precise spatial correspondence between the two signals in order to then be able to make a comparison of the two signals. So look forward to your questions. Yeah, thank you.
Right. Entirely my fault. We only have a couple of moments for a question. Sorry, Sarah. So does anyone have a question for Sarah before we move on?
So basically you're going to have two images with different contrast and you expect to reconstruct the same scene. At the end of the day, if they are different contrast, there are going to be differences. How much difference do you expect?
So when you say three different images, what do you mean?
Yeah, so no matter what we do, the physical principles and the physiological principles under which fMRI and the fNIRS work, they are different.
I see what you're saying.
For better or good, right? And so, there are going to be difference. How much difference are you expecting?
So here, I think we're thinking about prior work and the work that has already been done, looking at the correspondence is important. So there has already been other simultaneous studies that have looked at specific regions of interest, looking at the correspondence, and there is high correlation between the two signals. So what we expect to do is follow up on that work and not only do an ROI analysis, but also look at the whole brain level and see how that correlation may vary as a function of area of the cortex that we are sampling. So you're correct, and I think that that's something that has already been captured in prior literature as well. So we are kind of building on that and see what do we see when we are looking at the whole brain level, and also what happens in developmental populations as well. Yeah.
Very good, Liz. Thanks, Sarah, again. So next up is Morgan Fogarty. Come on up. Give me 1 second. So Morgan is going to be, where the slides are. Morgan is going to be discussing leveraging fMRI data to simulate and evaluate the potential performance of an fNIRS or DOT system.
Is it good? Okay. So thank you so much for the introduction and for the opportunity to share my work with you all today. So there are many decisions to be made when formulating a new experiment. What pad configuration covers the necessary brain regions, what stimulus design will be both engaging and elicit the desired activations, and how much data is required to achieve the necessary accuracy for your prediction models. These early experiment decisions are crucial to developing a successful new system or study design. And while we can use literature reviews to answer many of our questions, a method to quickly and easily evaluate system designs and their corresponding protocols prior to any data collection would better establish experiment feasibility. These methods can be made possible through the usage of Okay. So these methods can be made possible through the usage of publicly available fMRI data. So over the past decade, significant effort has been placed on open science, specifically in the context of open data. So barriers to data sharing, such as infrastructure and technical issues, legal barriers, and lack of organization and standards between research centers have been addressed by many experts in the field, and we continue to see policy and technology shifts that allow for even more accessible data. One primary example is the OpenNeuro platform, which was started in 2011 with much of the early data from Russ Poldrak's Open fMRI project. OpenNeuro has shaped the way that neuroimaging data is shared publicly, and the amount of data has steadily grown over the years. OpenNeuro has provided all researchers the opportunities to share their data, allowing for a wide range of ages, tasks, and quantity of data. Additionally, through harmonization efforts, large-scale projects such as the UK Biobank, the Human Connectome Project, ADNI, and many more provide a way to access thousands of subjects completing the same set of standardized tasks. As SfNIRS researchers, we can benefit from these fMRI data sharing advances and utilize these numerous datasets to gain insights into fNIRS study feasibility. Additionally, many of these datasets follow standards such as the BIDS data structure format, SIFTI formatting for the storage of connectivity data, and the HCP pre-processing pipeline. These standards across datasets allow us non-fMRI researchers to utilize these data easily. For functional MRI studies in particular, the rise of the usage of the Gordon cortical parcellation, as used in the HCP pipeline, has further moved the fMRI field towards more standardization. The Gordon parcellation separates the brain into 333 functionally derived cortical parcels, and these parcels can be further grouped into 13 functional networks. This is commonly used for these large-scale datasets to reduce file sizes by storing data in parcels as opposed to voxels. These data storage standards have provided more data accessibility and ease in using fMRI data. With the ever-growing number of publicly available datasets, we can assess studies with a wide range of subjects and tasks. So for instance, we can use precision studies. So for example, the Midnight Scan Club, which is collected at Washington University. And alternatively, we can use studies with limited sampling, but many subjects. So for example, the UK Biobank. This study contains more than 10,000 subjects, but with a very limited amount of fMRI data. And although it's a small amount per subject, the additional behavioral, neuropsychological, and physical assessments make this an incredibly rich data set. Both of these types of studies can provide important information for planning fNIRS systems and study designs. Large subject pools can be used for assessing pad configurations for specific brain networks or determining the amount of data required for tasks such as prediction. Alternatively, precision studies can be used to assess ideal stimulus designs for more niche tasks such as naturalistic viewing or decoding. So how can we use this extensive amount of fMRI data as fNIRS researchers? Since the endogenous contrast for the fMRI bold signal is also blood flow, we can use some of our existing data processing tools to simulate fNIRS measurements from the fMRI bold signal. We can think of this problem as simply as y equals ax, where we have the fMRI data, or x, and we're trying to solve for y, which is our simulated fNIRS measurements. To do this, we need to start by constructing our A matrix, or sensitivity matrix, which relates the relative changes in optical measurements at the surface to relative changes in absorption in the volume. This matrix can be generated for any fNIRS system using the NeuroDot toolbox. We start by generating a mesh from T1 and T2 structural data, which can either come for a particular subject or an atlas. And in this case, we're using the MNI atlas. And then we can generate a pad file for our system of interest, and we can place the optodes onto the mesh at the desired locations. From here, we can compute the sensitivity matrix by modeling the diffusion of photons through the head using NIRFast. And for more information on generating a pad file or an A matrix, I invite you to please check out our tutorial slides. So from this A matrix, we can begin by looking at the field of view of our imaging system and assess if we have the coverage needed for our desired study. So we can consider both the number of Gordon parcels within our field of view and the number of functional networks. So here we can do this comparison for one of our HD-DOT systems, and we can see that the system covers 109 parcels and 10 networks within our field of view. By assessing the number of visible parcels and networks and which parcels and networks are within our coverage, we can determine if a specific pad configuration is ideal for our intended study design. So for instance, if we're interested in studying cognitively demanding tasks, such as working memory or decision-making, we want to ensure full coverage of the frontal parietal network. Additionally, we can assess coverage based on the number of voxels within each of these 13 functional networks. So in this case, we can evaluate the field of view difference between a whole head fNIRS system and our ultra high density DOT system. So we can see that across 13 networks, we have a range of network visibility with the different fields of view, with each field of view covering at least 9 out of the 13 networks. So this type of simple analysis can provide quick intuitions regarding pad configuration to ensure the best field of view for the intended study, depending on which areas you're most interested in, as well as taking into consideration any logistical criteria for your study. So to better assess a study design or system configuration, we can follow our full simulation pipeline. So here we start with our fMRI bold signal, and in many cases, this will be formatted in the Gordon parcellation, which means our first step is to convert from parcel space to voxel space. And to do this, we generated and used a parcel to voxel matrix to get the bold data in a voxel by time format. Next, we can generate name matrix for our imaging system, and additionally, we can estimate a noise model to account for the optical noise of the system that's not present in the fMRI data. From there, we can take our A matrix, noise model, and the fMRI data to simulate our optical measurements through y equals ax plus n. And finally, we can reconstruct the measurements by first inverting and smoothing the A matrix, and then multiplying the inverted A matrix by our simulated measurements, resulting in our reconstructed simulated data. Then we can either do our task analysis, or in our case, we're going to do functional connectivity analysis in order to inform our system and stimulus design choices. So these methods were validated using subject-matched fMRI and DOT data from a previous iteration of our HD-DOT system, and this data actually comes from Adam Egebrecht's 2014 Nature Photonics paper. So here we can visually assess the similarities between the collected and simulated HD-DOT data for 3 functional connectivity seeds: motor, visual, and frontal parietal, with the seed locations denoted in dark green on the system field of view maps. We can see that these maps correspond to one another, as well as literature on functional connectivity, indicating that the simulation methods were successful. However, we want to also quantitatively assess the similarities. And to do so, we created overlap maps with the collected DOT data in blue, the simulated DOT data in pink, and the overlap in purple. We can see a substantial amount of overlap between these data, indicating that our simulation methods can produce results similar to collected DOT data. To further quantitatively assess the similarities, these functional connectivity maps are binarized and compared using the dice coefficient. So we can see that these dice coefficients are close to 1, which indicates that they are similar. So to determine the practicality of these methods, we analyzed the script using the MATLAB profiler tool to determine the computational load. And so this was all done on a Dell Precision desktop computer running Windows 10 with 64 gigs of RAM. So we were able to compute the total time and memory for simulating DOT data from a single fMRI dataset, computing the functional connectivity. So the process in total took about 2 minutes for HD-DOT system, as shown here, with the true core of our simulation methods, the y equals ax step, taking less than a second to compute, whereas the most time-consuming step was inverting the A matrix, which took around 80 seconds, but only needs to be computed once instead of per subject. So the one thing that isn't included in this is the generation of the A matrix and the pad file, and there have been some steps recently taken in order to have NeuroDoc compatible with Python and cloud computing resources. So for more information, I encourage you all to check out poster number 11 tomorrow. So overall, we wanted to show that these methods are computationally simple enough to be computed on a standard desktop workstation. So now quickly on to just one type of experimental design question that we can answer using simulated DOT. And so here we want to predict age from resting state connectivity data from the Healthy Brain Network project. And we can do a simple variation of our simulation workflow by limiting the field of view to our optical system field of view, as opposed to running the entire simulation pipeline. So the first step, this can be used just as a first step to establish proof of concept prior to simulating these large data sets. So starting with data from 428 children, we computed the functional connectivity matrix for the Gordon parcellation space. From there, we either used the full matrix, or we used only the parcels within the fNIRS or DOT field of view. And to do this, we retained a parcel if at least 50% of the voxels were within our optical field of view. Then the data was divided and trained using a linear support vector machine using 10-fold cross-validation to predict age. If we look at the results from the full Gordon parcellation, we can see that there's a strong agreement between the actual and predicted ages. But really, we want to see what this looks like in optical field of views. So here we can see the results for an example HD and UHD system. And we can see that while the results aren't as strong as the full Gordon parcellation, we can still achieve significance for both systems. And in particular, the UHD system achieves results that are very similar to the Gordon parcellation results. So overall, this shows that we can design and test different pad configurations to achieve our desired results for tasks such as age prediction, for instance, by targeting the parcels that have the greatest impact on the prediction results. After determining the ideal pad configuration, the system could then be built and used for this specific task. We can also use this information to determine factors such as the minimum number of participants required to make this age prediction method to achieve significance. So overall, these methods we can simulate optical data and assess system designs and study protocols prior to any optical data collection or even system development. The ability to simulate fNIRS measurements easily and efficiently from fMRI data allows us to make informed decisions during the design phase of a new experiment. And with the rise of large-scale publicly available fMRI data sets, we have access to an incredibly wide range of data and studies. And in all, the ability to leverage fMRI data to simulate and evaluate fNIRS system and study designs allows for a data-driven approach to making informed decisions about stimulus protocols and can assist us with the designing of new systems. So with that, I would like to thank everyone involved in this project as well as our funding sources. And I'd also like to highlight the other posters from the Culver Lab that are being presented this week. So thank you all for listening. I'm happy to take any questions.
Right. One single question, please. No? All right, I'll make one thing. What will be the advantage of fully synthetic data? Because at the end of the day, in fully synthetic data, I control the ground truth. Here, I don't control the ground truth. So what's the advantage?
So the advantage is really being able to kind of test these different studies out prior to any of our actual data collection. So we can simulate this data and look to see that we could get the results that we're interested in, and then go through and do our full data collection rather than going down either a rabbit hole of a stimulus design that might not work or developing a system that might not have the ideal coverage for our study. This is kind of like the first step that we can make for designing any new experiment or any new system.
Brilliant. Thank you so much, Morgan. All right, so let's thank again, Morgan. It is my pleasure to present an absolute veteran on this, which is Vlad Toronov and a hyperspectral analysis of the superficial changes cancellation using a short distance channel. Vlad, welcome.
Hello, everyone. Thank you for having me. So this work was inspired by clinical applications, and it is quite popular now to try to assess the cerebral vascular reactivity using paradigms such as hyperoxia, hypercapnia, breath holding, and these measurements can be done using functional MRI, of course. And of course, we want to do the same using fNIRS, but without the need to enjoying all benefits of near-infrared spectroscopy. But the problem is that in these paradigms, the changes occur not only in the brain, but in the entire body. And by the way, similar situation occurs during the cardiac surgery, and in particular, those surgeries when the surgeon must stop the heart for some periods of time. So the question we try to address is whether we can measure the response from the brain and specific cerebral response from the brain in this situation. So, and the problem is that in it already, actually this talk resonates with many others which are concerned with the extracerebral tissue influence on the fNIRS measurements, on the NIRS measurements. So the problem is that in NIRS we have to deal with the light propagation through different layers, and as I already said, in this situation, in all of these layers, not in all of them, but in at least in the vascularized layers, we can expect some response, and not necessarily only from the cortex. And on the other hand, it is known from the Monte Carlo simulations and from the theory that the high sensitivity in the single channel NIRS is right under the optodes. So, and as I said, we want to find out if we can find a specific cerebral response. We actually hope that the brain responds in at least in some way different than the peripheral tissue. So in our approach, we use the hyperspectral, or others call it broadband near-infrared spectroscopy, where we use just the light bulbs, such as the automotive headlight, for example. And the detector is actually a fiber optic spectrometer, and actually now there is a bunch of very highly sensitive spectrometers. And this system, we validated in invasive measurements on the pigs, where we could put the aptodes directly on the brain of the pig. But in these human measurements, of course, we couldn't do that. But we used the optical sensor, which had three source detector separations, 10, 30, and 40 millimeters. And assuming that the short distance channel interrogates only the scalp, and at 3 and 4 centimeters, we can somehow touch the cortex. And we use the breath holding paradigm with some long, actually, breath holdings of 30 seconds, half a minute, separated by 90 seconds of rest. So every subject would do 3 breath holds and then some baseline period. And this sensor was just attached to the subject's forehead, for left forehead. The chromophores we are targeting with the broadband hyperspectral are the changes in deoxyhemoglobin, oxyhemoglobin, and cytochrome C oxidase redox. And of course, we have water here, but we do not expect water to change, although it is included in our theoretical model. So the theoretical model, actually, sometimes we use the model based on the diffusion equation, analytical solutions to the diffusion equation, and it can be homogeneous model or layered models. And the way it works is we do just the pure fitting. We fit the experimental spectrum to the theoretical one predicted by the theory, and actually this is, we use the spectral derivatives as was explained yesterday in Mamadou Diop talk. So, and now the results are, I'm starting to show the results. So the top level shows just the amount of light, the spectral intensity measured at 1 centimeter, at 3 centimeters, and at 4 centimeters for different wavelengths. So what you can see here is that for 1 and 3 centimeters, the signal decreases where the water absorption is high, actually. But for 4 centimeters, the signal remains pretty high, even that way for those wavelengths. And how that can be? That can be because for 4 centimeters, we have channeling of light mainly through the skull, which has not that much water. Also, the next level shows here the cross-correlation of wavelength by wavelength cross-correlation, of temporal cross-correlation of responses to breath holdings. So here we cross-correlate 1 centimeter and 3 centimeter channels, 1 and 4 centimeters, and 3 centimeters and 4. So what we can see from this 1 and 3 centimeter cross correlation is that for all wavelengths, we have, for most of wavelengths, we have very high cross correlation, temporal cross correlation. That indicates that probably the responses in 3 centimeter channel are almost the same as in 1 centimeter. And then the cross correlation is slightly smaller lower between 4 centimeter and 1 centimeter channel, and relatively higher, but not for all wavelengths, between 3 and 4 centimeter channels. Now, again, in this, on top, I still show the spectral intensity, and the bottom row shows now the changes, the amplitude of changes at different wavelengths as a in response to the breath holding exercise. What we can see here is that actually at 1 centimeter and 3 centimeters, the amplitude of changes are also behave in a similar way. While for 4 centimeters, we have a decrease, strong decrease at longer wavelengths. And again, that can be, again, for the same reason that for which we have a lot of light at these wavelengths at the baseline, at the spectral intensity, because the light in this case is mostly propagating through the skull, and there is no change in the skull. So that means that actually we don't want to use these wavelengths higher than 900 nanometers in order to measure the cerebral response. So now we're still using the homogeneous model for a while, and these are the time traces for oxyhemoglobin on the top level, deoxyhemoglobin in the middle, and cytochrome C oxidase on the bottom at 1, 3, and 4 centimeters. And what we can actually see here that if we look closely, then we can see that actually for 1 and 3 centimeters, the temporal changes in oxy and deoxyhemoglobin are very similar for both 1 and 3 centimeters. For 4 centimeters, they are slightly different. And for cytochrome C oxidase, we actually don't see much responses at 1 and 3 centimeters, but we can see some at 4 centimeters. And in the next slide is seen better, where we compare now only 1 against 4 centimeter for oxyhemoglobin, deoxyhemoglobin, and cytochrome C oxidase. You can see that it is the actual response at 4 centimeters in cytochrome C oxidase, and then at the baseline we don't have it. So it's not just noise. So from this, actually, we could see that the changes at 1 and 3 centimeters in hemoglobin are almost the same, While in cytochrome C oxidase, we could see much higher changes in response to the breath holding at 4 centimeters than at 3 centimeters. So the next step was to analyze to apply the layered model, and the layered model was actually the modified Lambert-Beer's law for two-layered system, for two-layered structure. And what we did in this analysis, we did the temporal regression, the regression in the time domain of the time traces corresponding to 1 centimeter from long distance channels at 3 and 4 centimeters. And then the second step was to do the linear regression in spectral domain to obtain the changes in cytochrome C oxidase, oxy, and deoxyhemoglobin. And this is what you see in this slide. So you have changes in oxyhemoglobin after the regression, deoxyhemoglobin, and cytochrome C oxidase, and total hemoglobin. So what you can see that actually in the worst signal after regression is in oxyhemoglobin, and that shows that probably those are actually the curves here are when we subtract 1 centimeter out of 3, out of 4, or 3 out of 4. And the best signal we can see when we subtract 1 out of 3 actually. But also when we subtract 1 out of 4, there is just smaller amplitude but similar changes. But what we can see here is that oxyhemoglobin after this procedure is just gone, almost gone. So that means that oxyhemoglobin response measured by NIRS is actually, I mean, it's just the same as in the superficial tissue. So it's probably, there is no much difference in the brain response in terms of the oxyhemoglobin. However, in the deoxyhemoglobin, we see much stronger response after this procedure, in particular at 3 centimeters. Also, we see good response at 3 centimeters and cytochrome C oxidase, but also in 4 centimeters after subtracting the superficial. And so I'm almost done. So we can compare it with some fMRI studies of the time course. And in particular, our results better correspond. There was this nice paper, and our results better corresponds to the areas of the brain showing the bold signal, those areas which are irrigated by the middle cerebral artery. And that bold signal is actually here in the oxy, in this initial dip, which appears after this regression. So this initial dip corresponds to the increase in the bold signal. So the conclusions are that without using the short distance channel none of the chromophores measured at 3 centimeters clearly reflected cerebral signal or signal which is different from the superficial. At 4 centimeters, this separation, cerebral changes could be detected even without doing this regression. We could spot some differences in changes in deoxyhemoglobin and cytochrome C oxidase. But the specific cerebral response was separated from the extracerebral biasing using the time domain linear regression using the data from the short distance channel. The optimal waveband for the measurement of cytochrome C oxidase was between 750 and 900 nanometers. And of course, a lot of things we can do from this. We can now study this CVR, apply this method to study CVR, and actually study cytochrome C oxidase and various other related to the brain dynamics. And I would like to thank my Faculty of Science and St. Michael's Foundation Translational Innovation Fund for funding. And thank you very much for listening.
Well, thank you very much. Please, only one question because we are running out of time. Amaru, please.
Great talk, Vlad. So do you think that what you have seen, which is that when you have the signal, the oxy and deoxy is only when you regress the short data or distances that you have a good signal. Do you think that is that specific for breath hold, or that will propagate into other protocol?
Okay, actually, I think that our results are relevant not only to the breath holding. I think they are relevant to many features which are actually found in many fNIRS studies, not only breath hold, I think.
Right. Thank you very much, Professor Tornov. We thank you again. Right. We now move to our last speaker of the session from New York University, Shenghao Su, who is going to speak on multiverse analysis reliability index MARI to evaluate the reliability of fNIRS results. Shenhao, thank you. Welcome.
Okay, so hi everyone, and first I'm greatly honored to attend as an oral speaker. So my name is Zhou Chenghao, and currently I'm a master student from Department of Psychology at New York University. And today I will give you an example of how to evaluate the reliability of fNIRS results based on the multiverse insights. Okay, so this photo is shot 3 years ago when I was a sophomore year student. At that day, our college got our first fNIRS equipment, and after that, we have done a series of experiments with it. So we use hyperscanning and machine learning to decode the interpersonal touch on romantic lovers and friends. Well, but however, we have always been confusing about the parameters during the preprocessing analysis. What is this? It doesn't move. And yeah. Oh. Yeah, it works. Okay. And so the table on the left, it collected the range of the filters reported by some published papers in neuroimage in 2020 and 2021. So basically, no one used the same filtering parameters, which make me think in which degree does the filter range affect the data. And so when we set the cutoff threshold, our signal does not drop vertically, but it gradually decays. So when a low pass set was 0.3 and 0.31, so what is the differences between the effect of our experiment apart from the results? And another example is the right side of the table. It shows there are lots of technology to do the artificial rejections. So there are so many possibilities like we can do. And let me introduce what is the reliability in current study. So what's the significance of our nice results that we get from our raw data, apart from being maybe $700 in our favorite journal, or how is the reliability of it? So in today's presentation, and like I showed in the left side of the page, the reliability has been defined as the robustness that we have one data set and we have many similar pipelines, and we compare the distribution and the variabilities of our results. So this is also can be called like the multiverse exercise. The traditional exercise is to make some subjective choices and choose a reasonable path across the different exercise steps. So it is possible that such flexible exercise strategies will lead to different results and conclusions. And one solution is the pre-register that we use planet exercise. And I think for tomorrow's oral speaking, David will provide a guide and template on how to use the prostration as a tool for the transparent fNIRS study design. And let's go back to the multiverse exercise. So generally, it is a strategy that creates a list of model components which include all subject choices to generate different final data from the multi-workflows. So how to combine the multiverse exercise with our processing pipelines? As I showed in the left side, it's just like, and each possible choice in the process will create branches in these tree structures. And for the flow for the right side, like the word in the purple, it means the freedom choices. So, and after we have generated a multiverse, the next question is how to quantify the variability of these possibilities. So the following page is a description, and here we provide an index named the multiverse exercise reliability index. So it is to calculate the area under the red curve that covered by the first significant time point to the last significant time point. So it's essentially just take a rate, but now it looks very abstract. So I'm going to apply it to a data set step by step and show you how to compute it. So our data is from a picture viewing task to 2 groups of participants, and we got a total of 65 people, and they view 3 kinds of pictures and stimulate. So first is the broad oxygen type. So since they all have different physiological meanings, we decided to include all of them in our exercise. And the next is the bandpass filter. So we assume that a 0.01 disbalance in the frequency domain, it should not affect the result significantly. So we use a sliding window to generate a range of the bandpass filter. And the third is we include all time points in our universe instead of composing the time domain data to 1 point. The heat map on the right, on the left side shows like the unstable of the max, meaning, or mean approaches. So, by the way, so all exercise is based on the MAE package in Python, and this heat map is generated from the finger tagging task data, and it's on the website of the MAE. So, in summary, so this is the strategy that we use in our current study to generate the multiverse universe. And in the end, we generated a universe with 406,000 possibilities. And next, we are going to calculate the MAI index. Okay, so this is a demonstration of one single universe for each time point, and we can see it as a 1D vector. So based on the different process pipelines, we will get a matrix. The Y-ray is the different exercise pipeline, while the X-ray is time point. So since we have 2 groups of participants and 3 conditions, so we do an ANOVA test, and we correct the p-value, and in the end, we get a matrix of H Actually, it should be 3 matrices of which, because we have 2 mean effect and 1 interaction. And if the p-value is less than 0.001, so the h will be 1 and else it will be 0. Okay, so now we got a matrix. And first we find the first and last significant time points among all universes. And then between these 2 time points, we count the number of h across all universe for each time point, like across the X-ray. So across the X-ray, these numbers, it will make a dispersion curve, in red, the red curve. And then in the last step is to divide the area of red curve by the ideal square. And this area is made by dashed lines. It just means the time point multiplied all possibilities. So our index is a residual index, so it should range from 0 to 1. And the closer it is to 1, it means the universe like you generated, they all have the similar response. And so it shows like your results has a more reliability. And this is a heat map of the MRI in different conditions and in different countries. Oh, sorry, it's not in different countries, in different conditions. Okay, it's very
3 minutes.
Okay. So as we can see, this heat map shows that some of the response could be affected by the modern components like the channel 9's HB total and the channel 12's HBR and channel 15's HBO. So this significant results, it can be detected among some universe, but among the all universe, it was difficult to consider them as a reliable effect. And some of the significant results can be found in HBR with small robustness based on the MAI. And channel 1's HBO, it shows a relatively stable results that even if the different preprocessing parameters are used, results are close and significant in the starting position. And also the tendency is similar. But for HBR, for channel 3, this is unusual for NIRS data, even especially for the cognitive test, because the blood oxygen concentration, it should be relatively smooth without the phenomena shows in the green circle. It's too small. So, and next we provide a demonstration that visualizes the impact of each steps on the resulting data. So this is a distance matrix for the data from each universe. And first of all, we can see the HBO and HBR are far away from each other in group 1. And that is exactly what happens in between when you have an activation in certain brain area. And secondly, we found that in some cases, under the same condition, the distance of different HBR signal would be changed with the change of filters change. And for each step of preprocessing, we can always calculate the MRI results to see the influence of these steps. So why we do this? First, buyers may come with the freedom of researcher's degree in preprocess analysis. Although the data are collected objectively, but such freedom may bias your data due to the prior experience or unpredictable situations. And second, multiverse can help you. Like for example, according to the distance matrix, we change one thing at one time and we can see what happens. When we present the multiverse, a multiverse analysis can also raise awareness of how data flexibility can be exploited. And what does it mean if the results hang on it? And can we expect like these types of models to return the same answer? Or which conditions will they diverge? Does it make sense to expect the same results for these different pipelines? And what does it mean if the results are vary? So what we will do next is first, we will add the attribution reductions, and then we may verify the sensibility. And the last, we want to optimize this because it's a full permutation, so it may take a long time to calculate. And in current study, we assume that all universe, the weights are all equal to 1. But sometimes I don't think it makes sense, so we may change the weight of each universe. And thank you for listening. And it's an ongoing project. If you are interested, you can just send me my email, and it's chenhao.zhou@nyu.edu. Yeah, thank you. If you got questions, you can ask.
Okay, we have a couple minutes for questions.
Thank you for an interesting talk. Multiverse, that sounds really good. I can try a lot of stuff, but don't you impose this problem of multiple comparisons problem? So you're actually manipulating the data in different parameters and different dimensions, and you're reinforcing, maybe can you reinforce the false result because you're manipulating data? So I know it's based on, it looks like it's based on a p-value and statistics. And do you penalize based on multiple comparisons?
Yeah, we can add the models like I don't think the p-value is the only effect. Maybe we can use the effect size or other index. Yes.
Okay, thanks.
Any other questions?
Yeah.
Okay, I
Okay.
think
Thank you.
the copy's
Bye.
fine. Thank you so much. Thank you, everyone. We had multiple channels, multiple wavelengths, multiple modalities, and multiple universes. This was quite the session. So thank you, everyone. We'll have coffee for the next few minutes. I think we'll start at 4:05. Thank you.
Okay, so I think we have to start this last session on hardware. Please come in, sit down, enjoy and relax. Okay, this last session is on hardware. We have 1 invited talk and 5 contributed talk. We have to be very sharp because after the end we will go to the social event. So I ask the speaker to be very sharp.
Yeah, thank you. We had some very exciting talks this session. I'm glad you're all here. Our first speaker of the session will be Ashwin Parthasarathi from the University of South Florida. So thanks, Ashwin.
Thank you, Wes. Thank you, Wes, for that.
Hello, can you hear me? All right. Thank you, Wes, for the introduction, and thank you to the committee and the organizers for inviting me to give this talk. It's a pleasure to be here today to share our work on low-cost diffuse correlation spectroscopy devices with all of you in this nice conference. All right, so I want to first thank my funding sources, specifically the National Institute of Health and Southeastern Center for Electrical Engineering Education, and also disclose that some of these technologies that I'm talking about today I have intellectual property rights interests, and I also have equity stake in a startup company that's focusing on commercializing some of these technologies. So my lab at the University of South Florida is mainly focused on instrumentation development, primarily for biomedical optics. We focus on a variety of different applications. Clinically, we're interested in looking at brain injury monitoring with diffuse optical instruments. We're also interested in building newer devices, or pushing the technologies a little bit to make devices smaller, cheaper, and also make better measurements. Really, we like to do some imaging work, and we're also focusing on some interoperative and point-of-care measurements applications as well. But today I'm going to talk about this variable fiberless implementation of diffuse correlation spectroscopy. It's really the work of my grad student Sarinam Biswas, who's not here today because he's busy preparing for his PhD defense. So good luck to him in a few weeks. But our goal really is to create this variable CVF monitor that's about the size of a piece of gum, or maybe a little bit thicker than that, that can replace a big standard of care, or not standard of care, a big research grade diffuse correlation spectroscopy instruments. Really, our long-term vision, or kind of our goal, is to make these DCS devices as easy to use as a pulse ox. And if we can get there, it would make these devices a lot more accessible and easier to use, and really open up a lot of new applications for this technology. All right, so let's see how we can get to that position. This audience probably knows a little bit about diffuse correlation spectroscopy, but just to give a very quick overview, DCS is kind of an analog to NIRS. Instead of looking at, so it uses similar sources and detector arrangements. So it illuminate the tissue at a point, and then you detect the light at a different point, maybe a few, 2.5 centimeters or 3 centimeters away on the surface. But instead of looking at tissue absorption changes, DCS looks at the intensity fluctuations. So the time varying fluctuations of the intensity is what gives you information. And so to do that, DCS uses a coherent source like a laser, and basically when you have these coherent source light paths of different light paths pass through the tissue and they interfere at the detector, and this interference can be time varying and spatially varying depending on what kind of moving particles you have in the tissue. Whenever you have any kind of moving particle, the light paths, the path length of the light changes, and because of this change in path length, the intensity fluctuates in both time and space. So if you have a camera where you're detecting it, you would get like a speckle pattern, which is just this grainy pattern of, it's kind of like grainy pattern. It kind of looks like what your TV would look like when you lose power, when your satellite company loses power. That's kind of what it looks like. But if you basically look at it, if you basically put a point detector like a fiber, and then you detect the intensities very quickly, you get these fluctuating intensities. The thing is, you can quantify these intensities, the fluctuations very well by computing the intensity autocorrelation function and fit that to a diffusion model and get a quantitative index of blood flow. And this quantitative index of blood flow has been validated against TCD, has been validated against MRI and CT. So it's been very well tested to be a good measure of blood perfusion. So I want to talk about the typical instrument that's used for DCS. Now there are a variety of new changes, especially in the last few years. So especially time domain DCS or frequency domain or multi-distance or multi-wavelength DCS. I'm not focusing on that right now, but the conventional DCS instrument that we have typically used has a long coherence length laser, and this long coherence length laser is about $10,000 to $15,000 per device. And we've used long coherence length lasers because we want the coherence length needs to be high enough to account for the spread in light paths through tissue. And then you have your detectors, which we have typically used photon counting detectors. And these photon counting detector arrays can be about $10,000 for 4 channels. And the reason why we need these really high sensitivity detectors is you typically look at speckle at a single point, and you want to look at that single point because that gives you the best sensitivity. And because looking at a 5 micron point in tissue, basically you're collecting picowatts of light at that point. So to be able to get something detectable from it, you use photon counting. And then you have these counter electronics. This is the National Instrument System, but you can use FPGAs, and as many people have done. But anyway, either of those cost a little bit more money. And the bigger thing is that because all of these are fiber based, they end up being like a kind of a bulky instrument. And more important than that, especially if you want to scale this instrument for imaging, for example, you're going to need about like 64 of these because at every single detection point, let's say at 2.5 centimeter source detector separation, you need at least 4 detectors. So you're going to bundle in 4 fibers to a single detection point. And so as you can see, so as you can imagine, the scale of the instrument is going to like blow up very quickly if you want to do imaging with this. And so the alternative really then is to kind of go away with, move away from the traditional way or the conventional way we are doing it, to look at other alternative ways to make these make these blood flow measurements. So I'll talk about the source first. And so we basically built a fiberless source for making DCS. And basically, the main consideration for the source is the coherence length. And conventionally, we've used these long coherence length lasers. These are single frequency wavelength stabilized diode lasers, which have a coherence length of over 50 meters. And because we use long multimode illumination fibers, you kind of have to account for the coherence spread in the fibers as well as in addition to the spread in tissue. But if you think about it, the coherence length requirement itself is actually pretty small. It's actually, you just need your light source to have a coherence length greater than the spread of path lengths that you would see in tissue. And that works out very, very conservatively. That works out to be about 30 to 50 centimeters, which we can actually realize very easily with off-the-shelf diode. So that's what we built. So we took an off-the-shelf diode laser from Thor Labs, and then we tacked on a 3D printed part to collimate the diode. And we built this device, which is really small and portable that you can just kind of like hold on to your tissue or your head. And we compare and we tested out how it did compared to a wavelength stabilized source. So here's an example of autocorrelation functions that we measured from a liquid phantom for different source detector separations. So we get these DCS autocorrelation functions that are similar to what we would get with a conventional source. So the key here is, of course, that the source is directly on the sample and there's no fiber connecting to it. The detector here is still a regular conventional DCS detector. Okay, so we also wanted to check if this fiberless source produces accurate blood flow measurements. So we built a validation probe, which has a fiberless source, which is the red circle there, and then which has a conventional DCS source, which is your long coherence length wavelength stabilized diode. And then we made measurements at 1, 2.5, and 3.5 centimeter separations. I'm only going to show you the 1 and 2.5 centimeter separations, but we can look at our paper for the 3.5 centimeter separations. And as I'll show in a short while, the sources are really comparable, right? So in this experiment, again, this was a measurement on a liquid phantom. We just measured the blood flow index at these two source detector separations over time, and then we plotted them out. So the blue curve is your wavelength stabilized source. The red curve is your fiberless source. And the scatter plot also gives you kind of the spread of the blood flow indices that we measure. But on average, the blood flow index that we measure with the fiberless source is pretty similar to the one that you measure with the wavelength stabilized source. In fact, there's no statistically significant difference between the two measurements. So that's your take-home message there. All right, so then more importantly, we wanted to characterize the signal to noise ratio and the noise of these measurements. So we did a controlled liquid phantom experiment again, where we control the intensity of the laser to get different detection levels. And basically we track the noise and the signal to noise of the intensity autocorrelation functions. So these two curves on the left here, the two curves on the left are the noise of the DCS intensity autocorrelation function measured at a specific delay time. The middle two curves are the signal to noise ratio of the same DCS intensity autocorrelation functions. The individual points are the measurements, and then the solid lines are actually fits to a correlation noise model. And we found that, so basically we can see that the model really fits well. So basically your fiberless source works as well as a wavelength stabilized source. We in fact did a head-to-head comparison of the noise and the signal to noise ratio. Those are your curves on the right there. So the blue curves are the noise and the signal to noise ratio of your wavelength stabilized source. And the red curves are the noise and signal to noise ratio of the fiberless source. So basically, when you're comparing, if you're able to control for the intensity levels and the integration times, then you can get away with using a fiberless, little diode laser that you can get off Thor Labs for like $100, right? So that's our take-home message: is that the noise in the SNR of these fiberless sources is comparable to a wavelength stabilized source. So we were excited about this, and we wanted to do some in vivo experiments. So here's an experiment that we did we want to do an arm cuff occlusion, but this is just the baseline data of the source of the blood flow measured at 1 centimeter and 2.5 centimeters. So we see that the fiberless source has enough sensitivity to pick up fluctuations in your fluctuations in your blood flow. That kind of indicates that there's good sensitivity in the measurements. We also did the occlusion study. So the left two curves are the blood flow indices, and the right two curves are the relative blood flow changes. So both the fiberless and the wavelength stabilized source track the blood flow changes due to the occlusion. You see a good recovery, and the overshoot when your occlusion is recovered, the hyperemic response is also tracked. So this basically shows you that the dynamic range of the measurements are also pretty good. And so basically what we can conclude from all of this is that you can replace your wavelength stabilized source with a little diode laser as long as you don't use a fiber and put it directly onto the tissue. All right, so now what do we do about the detector? So the detector is a little bit more challenging. Conventionally, we've been using single photon counting detectors. Obviously, single photon counting detectors are not really conducive for variable measurements. It's you can't have a block detector on your head. And so we need to think of a different way to do this. And our approach is to use spectral contrast for this. And I should say that we're not the only ones who are using spectral contrast for these measurements. In fact, I think there's going to be a talk later on in this session about it. and Turgut's group has been using spectral contrast spectroscopy as well. Our approach is slightly different. Typically, spectral contrast spectroscopy measurements use a camera, and I'll actually talk about that in a little bit. But instead, we are using a generic photodiode, and this photodiode is something that I got out of DigiKey for like $10. The advantage is that you're not doing photon counting anymore. Instead, you're looking at the fluctuations of integrated intensity, and I'll talk about what I mean by that. But of course, the advantage is that it's a really small detector, and it's really a low cost and low noise measurement. Okay, so just to kind of give you an overview of how the spectral contrast type measurements work, I want to introduce you to laser speckle imaging, which is kind of a single scattering analog of DCS in many ways. So in laser speckle imaging, you just image the tissue image whatever tissue sample you have with diffuse laser illumination. Again, the single mode coherent illumination. And this is kind of the spectral pattern that I kind of alluded to in the beginning of my talk. So this is kind of the grainy pattern that you get that spatially tells you how your intensities are fluctuating. But it's actually a dynamic signal. Now you can actually compute blood flow by quite simply computing the spectral contrast. And the spectral contrast is just the ratio of the standard deviation of the pixels inside any kind of window to the mean of the pixels inside that window. And this window can be either a spatial window or a temporal window, or you can do some kind of mix and match. You can do a spatial temporal window as well. But the general idea is that each of these individual measurements, so to speak, are integrated intensities. So they are intensities that are being averaged by the integration time of your camera. So when I was a student, when I was a PhD student, I actually worked on a device to build what's called a multi-exposure instrument. And this multi-exposure instrument experimentally modulated the experimentally changed the exposure time of your camera to capture these speckle visibility curves. And the speckle visibility curves are analogous to the DCS intensity autocorrelation curves that you saw earlier. The difference being that these are now captured from integrated intensities. So at each point, the speckle visibility gives you the ratio of the standard deviation to the mean of speckle intensities. And these intensities are averaged over the integration time of your camera. So in this experiment, we change the exposure time of the camera or the integration time of the camera experimentally to get these curves. But the point is that if you're able to acquire these speckle visibility curves, you can fit that to a model that you like that's appropriate to your geometry, and then get blood flow again. But the obvious advantage here is that this doesn't use photon counting, but you just need to change the way you're detecting the light a little bit. So this is what we came up with. So instead of using a camera, we use a really simple unbiased photodiode. So this photodiode works in a photovoltaic mode. So that means it's not really biased. So there's no amplification of light that goes on in the photodiode. And so it has the lowest dark noise possible that you can get. And the light that you detect, or the current that you're detecting from the photodiode, we mimic the camera operation, so to speak, by using an op-amp integrator. This integrator is a programmable integrator. It has a switch in the feedback circuit. When the switch is open, the integrator gives an output that's proportional to the integral of the intensity of is proportional to the integral of the current that the photodiode is detecting. And then when you want to reset the circuit, you close the circuit. When you close the circuit, the capacitor in your feedback loop discharges, and the circuit is ready to operate again. And by basically, flipping this switch back and forth, and you can do this switch back and forth very easily using like a high quality BJT switch or a transistor switch. By flipping the switch back and forth, you can get these multiple integrated intensity outputs. So I'm going to show you a little demo. But before that, I want to also talk about one more thing. The other thing that we can do is we can actually control the amount of light that you're detecting by adding like a current source or a drain to the measurement point. Now, this is important because the detector that we're using is not your single photon, is not your single speckle detection. It's actually an extended measurement. And so that means that there's a lot of speckle averaging that happens in your detector. So you're going to wash out some of your fluctuations. So one simple way to kind of like overcome that is we just connect a programmable drain current that kind of that allows some of the average current to go to ground, so to speak. And then your integrator now basically integrates the fluctuating component of your current. And so it's kind of a quick and easy way to boost the contrast of your measurement. So here's kind of the workflow of what we do. So you collect these integrated intensity curves over, you collect several of these integrated intensity curves, and at any given exposure time, you can calculate the speckle contrast, again, by just doing the standard deviation to the mean of the intensities. And that gives you your speckle visibility curves here. You fit that to a diffuse speckle model now because we're looking at deep tissue measurements, and then that to estimate blood flow. So here's kind of so the advantage, of course, is you can do really fast measurements, and because you have really low dark noise, you can also measure the dark noise by just looking at the zero integration measurement and correct for it. And you can, and you know, depending on how many frames you use, you can get up to like 1 kilohertz data rate. So here is an example. I hope this is clearly visible for you. This is just like a screenshot of our LabVIEW interface software. So the figure on the left is the intensity as a function of time, of integration time, and the figure on the right is your speckle visibility curve. So this is real-time computation. And I believe the speckle visibility curves are computed with an average of about 100 raw frames. And you can actually appreciate, I like to show this because you can actually appreciate where your signal is coming from. Your signal is coming from the way in which your integrated intensity is kind of bouncing back and forth. So it's really the variation of the intensities at every single point that you're interested, that's giving you your signal. And these curves actually like kind of are really like almost DCS-like because the integration times are really small, but you know, obviously we need to use a diffuse speckle model. All right, so we've done some experiments on this. We're still optimizing the drain current portion of it a little bit, but we've done some preliminary experiments of it, which I'd like to share with you. So we did an arm cuff occlusion measurement on a healthy volunteer. So in this case, you know, it's a standard measurement where you place a probe on your forearm and then you occlude the blood flow there. So in the baseline, we're able to pick up the pulsatility, and this pulsatility corresponds to like a 1 hertz peak from your blood pressure. You're able to track the flow changes during the occlusion as well. You know, some other measures. So we are able to resolve pulsatility in some cases, and we are able to track the occlusion trends as well. So we also did a head-to-head comparison. So in this case, we had both the integrated DCS curve and the DCS curve together. And in this case, you can kind of see that the occlusion, there's kind of like a ceiling effect, so to speak. So we need to do some improvements over there. But you can still see that the trends are still maintained, and then the recovery is you're able to track the recovery as well. So we did a bilateral cuff occlusion measurement as well. So in this case, these are really small changes in blood pressure blood flow. So in this case, we inflated we placed 2 thigh cuffs on a volunteer, and then we inflated the cuffs and then deflated them suddenly. And when you deflate them, there's a momentary drop in blood pressure, and then that causes a momentary drop in blood flow. And we're able to track that in both your DCS and the IDCS curves. The IDCS curves are a little bit more noisier, but you know, that's again, like, these are a little preliminary. And we also did a breath hold experiment. Again, this is just measurement on one subject on 5 trials. So this is holding your breath for about 20 seconds after an exhalation, and the measurements with DCS and IDCS kind of like are correlated with each other, but the magnitudes are still a little bit off. So anyway, we still have a little bit more work to do to fine-tune the sensitivity. I believe that the characterizing the sensitivity of the device is going to be important in the future, which is something that we are working on right now. All right, in summary, I've shown you that we can build a deep tissue blood flow measurement device that costs, you know, maybe $2,200 to $500. You can use generic off-the-shelf parts using a custom integrator and a custom circuit to control for your speckle averaging. So there are a lot of applications for this, long-term monitoring, imaging applications like I talked about earlier, monitoring in challenging applications like pediatric conditions, peripheral flow monitoring, and much more. So with that, I'd like to thank my students who have worked on this project, and my collaborators as well. I'm happy to take any questions. Thank you.
Any questions? If you can come to the mic, that'd be great. And
Thank you, Ashwin. So, that's two questions. For the laser, that is awesome. That if we can use that, so you don't even need optical attenuator things. The light doesn't go back and it destabilizes.
No, you mean an isolator?
Yeah,
no, we don't really use an isolator. I mean, it's again like an isolator is something that I think an isolator is more critical if you're using single frequency.
Yeah.
Because the coherence length is long enough. That's one reason. The other reason I think is that really the coherence length needs to be long enough within the time frame of your measurement. So it can jump a little bit. The wavelength can jump a little bit, but as long as it's single frequency operation within the time frame of your measurement, which is a few microseconds, then I think it's still okay. I think that's one of the other reasons why we think we don't need an isolator.
I mean, so you get the same beta that we get with a long coherence?
Yes, yeah.
Okay. Yeah. And for the detector, yeah, there is more work there, I guess.
Yes, right. Yes, I agree. There's a little bit more work there to kind of fine tune the sensitivity.
So what I learned with this SCOS is that you have to do a lot of subtractions because otherwise it looks like intensity. Are you applying a subtraction of the various noise on these detectors?
So the only subtraction we do is the dark noise correction.
Yeah,
there's no other, I mean, the other noise is not significant because like in a camera, you have a lot of other noise factors that you come in because of the biasing and because of the readout noise and all that. Because we're just using an unbiased diode, our noise floor is really low. Like inherently it's low.
Okay.
So
it just doesn't look like the pulsatile doesn't look
the pulsatile doesn't look. Yeah, the pulsatile doesn't look really good, so it still needs a little bit of fine tuning. But I think there are two things that we could do over there. One is, but I think the speckle averaging takes a lot of pulls a lot of the sensitivity away from it, because I think it really depends on how much your curve can fluctuate with blood flow changes.
Got it.
Right. So we have tested, I didn't show this data, but we have tested looking at smaller detectors. And when you do look at smaller detectors, our variance does increase. So smaller detector areas or putting a pinhole in front of your detector area.
But then the sensitivity will go down.
Right, but
the photon efficiency.
The measurement, but in most of these cases, we're not light starved. I mean, like we still have a lot of measurements. Like in the demo that I showed you, we actually had to drain a lot of current and we were still able to get, like without draining the current, we were like almost saturating it.
Cool.
But I think that the advantage, that's because of the large detection area. So I think there's like a trade-off over there to be worked out.
Thank you.
Thank you.
Hi, you mentioned that you were having some challenges with the dark current draining approach that you're doing, and obviously you're doing this and you've got to do it as quiet as the photodiode is, which is probably quite challenging. Have you considered using like a quadrant photodetector or a dual element photodetector and actually removing that average, assuming that you're on a reasonably homogeneous area of the tissue optically?
I'm sorry, so can you repeat that?
Have you considered, instead of electronically removing that dark current, that average current, having something like a quadrant photodetector or a dual element photodetector? And then you could do that right locally at the tissue, wire that up. So you're subtracting your average current before you even do your, so you don't have to move it with a current sink later on.
Well, actually we are doing that with the current sink that we're doing is before we detect it.
Yeah.
I mean,
it's still implemented electronically though, right? You're having to sync that out at the input to your.
Yeah, so what we do is we have a current source. We have a controllable current source that we just tune while we're doing the measurement.
Right.
So it's not a post hoc correction.
No, sure. But if instead of doing that, you were to have essentially two photodiodes on the tissue and to subtract the photocurrents
you could do
tissue,
that, right?
Because then you don't have to adjust that. It's automatically going to remove the average
you could
current.
do that. I mean, yeah, that's right. You could do that too.
Okay.
Yeah.
But you haven't.
We haven't looked at that, but that's a good idea.
Just purely from the perspective of maximizing your dynamic range, it would automatically correct and get rid of that.
So like looking at like an array, like a 2x2 array or something.
Yeah, I mean you can get them as position sensors. Typically, they're used for, aren't they? So they're available.
That's a good idea. I mean that would work. That should work. Yeah.
Yeah. Okay. Thank you.
One just really quick question, Ashwin. Yeah, very exciting. Like I think the multi-exposure is a great idea. Did you compare the BFI with the multi-exposure to just using a single exposure time for the detector to see if that had a big benefit?
Oh, that's a good question. We haven't compared it to a single, we've been focused on comparing it with DCS measurements. We haven't compared that to a single exposure measurement. No, that's.
Yeah, this might be interesting to show, like if even the single, maybe how well that would agree with the DCS or the multi really brings a big.
Yeah, that's a good question. Yeah, that's a good point. I mean, we haven't looked at the single exposure because we've been focusing on doing the fit. I mean, with speckle, single versus multi-exposure is better for the quantitativeness, quantitative blood flow measurement. So we're kind of like going off that assumption, but maybe it's different here.
Okay.
Yeah.
Well, thanks so much, Ashwin. A wonderful
Thank you very
talk.
much.
Okay, so the next speaker is Luca Pollonini from University of Houston. Luca will speak about wearable, high density, fNIRS, EEG. Please.
Okay. Thank you very much, Alessandro, for the introduction. Good afternoon, everyone. So good to be here and see everyone after such a long time. So I will be presenting a project that describes the design of a multimodal instrument that I'm developing as a collaboration between my lab and the company Indus Instruments in the Houston area, and we're both part of the NSF-funded IUCRC Brain Center for the Development of Neurotechnologies. So as we all know, that's the reason we are here. fNIRS research is growing exponentially, but the same is true also for research in which fNIRS and EEG technologies are combined together. Of course, they offer complementary features that we all want to exploit as researchers. So there's a motivation for trying to combine these two technologies. But of relevance to my project here is that the same going trend is clearly observable already also for wearable fNIRS, and there are great examples also in the exhibit gallery of wearable technologies, and also we're starting to see an upgoing trend also for wearable fNIRS and EEG. So as Luisa Gosset pointed out very timely in her presentation yesterday, there is a desire by fNIRS researchers to transition from the typical, I would say, combined fNIRS and EEG instrumentation in which that unfortunately results in a headgear that is pretty bulky and heavyweight, not so easy to handle, to fully integrated fNIRS and EEG. And von Luhmann and Lee, back in 2017, and with their co-authors in IEEE papers, they've already pioneered this kind of integration. And more so, Alex, in his recent 2011 vision paper, as part of the NINJA-NIRS project, have pointed out how important the integration of the technologies, fNIRS, EEG, and other modalities as well, is being very important for the future. So in this area, we intend to contribute by developing an fNIRS and EEG instrument that is whole head coverage, wearable, to enable naturalistic studies. Now, from an instrument design perspective, of course, EEG and fNIRS, they have a lot of commonalities. Both of them require a good contact, a good coupling between the electrodes and the optode and the scalp to get good readings. That's especially true for dry EEG. That is very intolerant to poor coupling between the electrode and the scalp. And of course, both modalities benefit from ergonomic and lightweight headgears. However, there are other aspects in which fNIRS and EEG have a little bit more, let's say, divergent or contrasting requirements. And one of them is actually the layout of optodes and electrodes, because In fNIRS, the majorities of the studies, at least in regular fNIRS, maybe not so much in DOT and HD DOT, are regularly, the outputs are regularly spaced, and that's to achieve a fairly uniform interrogation of the cerebral cortex. That is not the case with EEG research, in which the investigators, they rely pretty heavily on 1010 layouts and 1020 layouts, and that are not, in which the distance between the electrodes are not regularly spaced. And the other aspect in which the two technologies contrast quite significantly is actually the cap, because if for fNIRS and DOT, is desirable to have a cap that is closed and dark to create a nice dark environment underneath the cap. For EEG, there is actually the preference of having a mesh-like or perspiring cap to avoid sweat artifacts. So our proposed solution to kind of find a compromise between all these requirements is to create an integrated fNIRS EEG node that consists of or that embeds one power adjustable dual wavelength LED, which is in the central emitter that you can see in this layout right there. Oh, you can see it clearly visibly. And that is surrounded by 4 photodetectors at a 10 millimeter distance to achieve a nice short separation measurement. And all of these elements are optically coupled to jacketed light pipes to achieve a sufficient penetration of hair. The EEG electrodes are consist in 4 or more 2 millimeter diameter metallic electrodes, which are terminated with some silver chloride ink for a good to achieve good scalp impedance. And we integrated, or we packaged all the electronics into a cubic opaque enclosure, which is approximately 20 by 20 by 20 millimeters, weighing approximately 5 grams. And we devised also a socket to host the integrated node that allows also for a vertical adjustment of approximately 10 millimeter in range, and also to interchange the prong length, choosing between 1, 5, and 10 millimeters to be able to be more diverse in our research and achieve hair penetration more or less regardless of hairstyle and hair types, or at least to the best of our ability. This is the block diagram of the integrated node. Each node integrates a microcontroller unit that allows us to either programmatically or automatically set the LED intensity at the 2 wavelengths separately. Of course, the unit is in charge for detecting light from all 4 photodiodes, and also from the EEG electrode. The reference for the EEG is actually is common to all nodes that are connected in the system. And the synchronization and communication between all the integrated nodes is achieved through a flexible, very lightweight PCB bus. And of course, all the firmware that is programmed into the nodes allows for a time multiplexed collection of short separation and long separation fNIRS measurements as well. So in terms of layout, we decided to make the EEG people happy by going with the 1010 layout. And this essentially results in a system that has 61 optical emitters, 244 detectors, and 61 electrodes in total. And you can see the picture of the layout here. In terms of fNIRS channels, we have a distribution that is depicted in this central histogram right here. Excuse me. We have approximately 1,200 long separation channels ranging between 25 and 50 millimeters. And you can also take a look at the sensitivity map. And you can clearly notice how the sensitivity is higher, or at least relatively higher, in the inferior part of the brain, because that's where the 10-10 layout placement is denser compared to the superior part. In terms of sampling rates, we can achieve 600 Hertz maximum sampling rate for EEG, 10 Hertz for fNIRS for the whole head scan. And we're implementing, data saving, streaming, and data organization according to the latest SNIRF, BIDS, and LSL standards. So this past summer, we started to implement our first prototype. Unfortunately, pretty much like everybody else, we got hit by a pretty severe chip shortage that slowed us down. So while we were waiting for the electronic components, we put together a full mock prototype of the system, because we wanted to ensure that this pretty dense and potentially pokey headgear would not bother significantly our future participants, especially the pediatric population. So we recruited a sample of pediatric participants across different ages. And we asked them essentially to wear the device for approximately 60 minutes and to essentially observe them and ask them to, report discomfort and whatnot. And the results were definitely positive. We got, good achievement, no complaints whatsoever about the weight. And by the way, the overall headgear is approximately a pound, slightly more or slightly less, depending on the thickness of the neoprene cap, and also the electronics inside. And full disclosure, this is my daughter. She did receive a good toy for taking this smiley picture after that. But it was well worth, because we were able also to convince the NIH that wearing this cap for a fairly long experiment is feasible in the pediatric population. And in the next 5 years, we will be studying and deploying the device to study developmental disabilities in the pediatric population in Zambia on a very large cohort of patients. I want to thank my collaborator and the PI of the grant, Elena Grigorenko, which is with the University of Houston. A few weeks ago, finally, we received all the electronics that we needed, and we started to do sanity checks and testing of our system, at least on a few nodes. We started to collect data to observe whether we achieved a fairly reasonable fall-off curve, a fairly typical fall-off curve that we can observe right here. And all the channels were measured above the dark measurements, or the noise floor, between 10 and 47 millimeters approximately. We also did take a look at what happened down at the noise floor. Especially, we were worrying about possible light piping with the LED firing off a fairly high intensity for long separation measurement. We wanted to ensure that the nearby, or the photodetectors that are within the same node would not actually take any reading above the noise floor. And you can see on the right, it doesn't really make a difference whether the LEDs are on or off. The readings are pretty much where we expect them to be. We did test also the EEG performance. We fed a 5 Hertz sine wave into the circuit. This plot essentially shows 3 different amplitudes, 1, 2, and 5 microvolts. And to give you an idea, typically the human EEG, depending on the frequency, but typical EEG amplitudes are a few tenths of microvolts. We can clearly see the sinusoidal or the quasi-sinusoidal waveform. So we're pretty happy with the SNR they were achieving thus far. We also checked that the switching on and off of the LEDs or other switching in the electronics would not compromise the EEG readouts. And this is the same sine wave that is collected through EEG that is not artifacted in any way by the fNIRS readouts, which are where the round markers are. We started to do also some preliminary in vivo testing, which are as fresh as this past week. We can read out EEG eye blinks without a problem. That's again a fairly good sanity check for EEG. And we could also have pretty prominent cardiac pulsation at long separation measurements for the fNIRS component. So I truly wish I could show you a little bit more functional activation, but unfortunately we ran out of time. We hope to report these measurements really soon. So I want to finally acknowledge Sridhar Madala and his team at Indus Instruments for this great collaboration. The NSF and NIH that are funding this research, and also the US Fulbright Commission and Fulbright España for sponsoring my sabbatical Spain at BCVL, where I seek to validate this device on language task in naturalistic environment. Thank you very much.
Thank you. We have time for one quick question. So I have one. So you said you made the EEG people happy choosing the 1020 system, but then you relax on the regular grid of fNIRS, so the sensitivity decreased. So do you think this is the right approach?
Well, if you take a look, the sensitivity map, there is a little bit less sensitivity in the upper area here, but it's not dramatically different, or is not. It doesn't become we're not totally blind to the cortical hemodynamics there. We discussed with a lot of EEG experts, and fundamentally they were not willing to compromise on the 1010 layout. Not very tolerant. I apologize to EEG people in the audience, but
okay. Thanks, Luca. I think we have to move to the next speaker. The next speaker is Guy Perkins from University of Birmingham. He will talk on utilization of frequency domain fNIRS to improve contrast.
Cool, thank you. Yeah, I hope everyone's feeling awake. So yeah, I'm talking today about frequency domain fNIRS. So let's get straight into it. So for those of you who don't know, what is frequency domain fNIRS? So you're probably all very familiar with continuous wave and time domain NIRS, but with frequency domain, essentially we have our light source, which is sinusoidally modulated at a modulation frequency omega, and essentially the light passes through the tissue, and then the light we measure has its amplitude reduced, and also the phase of that modulation has been shifted, so we measure our intensity and phase. And this talk is about looking at that phase and also the modulation frequency. So why investigate phase? So building on previous studies, we know that phase samples deeper than intensity, which can be demonstrated by looking at the sensitivity in the top row at two different source detector separations. As well as this, we know that phase has a more distributed sensitivity away from the surface. So what did we do to investigate this? So essentially, we had a very simple motor cortex stimulus where people had a series of left and right grips with a randomized rest period, and then a random stimulus order to the subject. We also did 3D camera registration to register the position of the optodes onto an atlas, and also to get our source detector separations. So we had to think very carefully about how we arranged our sources and detectors because of our hardware we were using. And the main thing was really, because our detectors are photomultiplier tubes, it means that if you have a light source close to it, it can saturate the detector, and therefore the gain or the voltage bias on the detector has to come down to accommodate for that. And this can then make it harder to do measurements from further away. So you'll notice there are two rows of detectors on the top and the bottom, and these facilitate measurements from 27 to about 37 millimeters. We also wanted to implement short channel regression. This was done at 9 millimeters by just plonking two detectors like so. We also wanted to make sure there were spatially overlapping channels to facilitate tomography. And then also, I thought it'd be very interesting to use some other techniques by different groups. So firstly, we looked at using the dual slopes method. If you don't know about that, I'd recommend reading up on it. It's very interesting. And then secondly, there's another method of analysis, which essentially you have the sources and detectors, two measurements in like a cross shape. If you multiply the Jacobian or the sensitivity of those, where there's no overlap, the sensitivity cancels out. And where there's overlap, you'll get a buildup of sensitivity, which is a bit deeper in the tissue. Okay, so what are the results basically? So we're just looking at like two regions of interest, 1 and 2. Region of interest 1 is where we expect our motor cortex activity. And you can see for the intensity, you get a pretty standard HRF. Then when we use our phase data, you get a slightly larger response in both oxy and deoxy. And then we've actually combined the intensity and phase together for simultaneous reconstruction. And the reason why we did this is if you look at the unilateral response, which is the dashed line, there's actually a bit of an artifact in the phase data. And typically the phase data is inherently noisier than the intensity. So hopefully by combining both these data types, we can pull the best attributes from both of them. And then finally, using the phase data, you can also account for changes in scattering. However, again, this is quite preliminary, and I'm not going to draw any bold conclusions from that. And then also, just to demonstrate, this is at the bottom without any short channel regression. And you can see that the intensity data, the HRF from that changes more when we do our regression than the phase, which may suggest, again, that the phase is less affected by superficial changes. And yeah, here's just our proof of concept with our intensity data only. So on the left, you just have these two source sector pairs, and we've just averaged the data and the Jacobian, and that's the response you get. In the middle, you have the 90-degree multiplication method, and then on the right-hand side, you have the dual slopes method. So it's reassuring that these three different approaches yield similar response functions. And again, I think ideally you would like repeat this and yeah, see how it varies for different stimulus and stuff. Okay, so the second part of my talk is looking at modulation frequency. So why investigate modulation frequency? So I've pulled a figure here from a paper, and if you haven't looked at this, are interested in fNIRS, like definitely check it out. So essentially, what this is saying is that if you vary the modulation frequency, and this was done in simulations, then you'll actually get an appreciable difference in the sensitivity as a function of depth with your phase data. And you'll get very small changes in sensitivity from the intensity data, which would suggest that, yeah, looking at the phase at different frequencies is an interesting thing to do. So what did we do? So very kindly, we have this phantom, the Neuroc phantom from Milan, and essentially we plonked our sources and detectors on top in the high density arrangement. And then in this phantom, there was this rod, and then in the rod, there's like a contrast anomaly. And this sits 15 millimeters deep in the phantom. And essentially, you can move the rod about to take measurements with the contrast present and the contrast not present. So what does that look like? So this is an interesting graph because basically the top row shows the intensity data at a typical nearest neighbor 1 and 2 measurement. And what you can see is you can clearly see like the baseline without the contrast. When the contrast is present, the change in data you can see. But what's most interesting is that if you look at the three colors, the blue, the pink, and the orange, they represent different modulation frequencies. And essentially, you get differences in the contrast as a function of modulation frequency. And for the phase, as the modulation frequency increases, you do get a bigger contrast. However, as you can visually see, the SNR also changes as well. So we did these measurements and did our tomography, and then we borrowed these evaluation metrics from the White and Culver paper. And yeah, these are our reconstructions. So these are just slices, but we did reconstruct in 3 dimensions. And essentially, the 2 things to note are that when we used our intensity only data, which I'm going to call like CW, that's the response we got. However, using our intensity and phase, we could see a bigger contrast for the anomaly, and then actually putting together the phase data at different modulation frequencies, this seems to reconstruct the blob better. However, we should note that there are artifacts present across both the CW and FD and multi-frequency cases. However, for the intensity case, this artifact is more in the Z direction, whereas for the frequency domain and MFD case, this was more in X and Y, which is probably representative of the fact that there's less depth sensitivity of intensity, but then you have the noise in the phase. Yeah, and then this is basically just showing a table of the metrics. So you've got like 3 different rods of different absorption, essentially, or effective absorption from different volumes of the anomaly. And essentially, we saw that the best localization, so a lower number here is better. The best localization in all 3 rods was from the multi-frequency data. The best, like 4.5 maximum, in 2 of the 3 cases came from either the FD or the MFD case. And then the best effective resolution came from the FD or MFD case. So yeah, what are the future directions? So essentially, it's all well and good that we've done this on a phantom, but clearly at some point you should, try and actually go on a real person. So I'd like to utilize this in vivo and essentially see if these benefits in phantom actually translate like to the real world. And doing this by incorporating the phase measurements and multi-frequency measurements. And if you're sat in a room and you're planning on constructing a frequency domain device, I'd highly strongly suggest that you can make it do simultaneous multi-frequency measurements. And I know there's a few different projects happening with that, because essentially, if you could tune your modulation frequency, then this will essentially like affect the tissue you're sampling. In this image, that's not to scale, that was just for hyperbole. So don't go thinking that you're going to get to the middle of the brain. So yeah, so what are the conclusions? So essentially, I thought this was a very nice analogy that if you're using just one modulation frequency in your fNIRS, then you've got this tug of war essentially between the contrast you're going to get from your intensity versus the contrast you get from your phase. However, you have to be very careful because you need to balance this with the changing SNR, and you need to consider the SNR of the phase and the intensity at the modulation frequency. However, if you do multi-frequency measurements, then essentially you can pull together different benefits and really try and understand all your trade-offs, and hopefully, incorporate the best of the phase contrast, incorporate the best of the SNR, the intensity contrast. You'll have different choices of frequencies because like different frequencies may work better for different subjects in different conditions, and you'll have more data to analyze. So yeah, thank you very much for listening. Big thank you to everyone that's helped me and stuff, especially Hamid, Felipe, and Adam, and my colleague Robin and Mario have posters, so you should definitely look at them. You can contact me on Twitter, ResearchGate, send me an email, or just like pull me for a chat. So thank you very much.
Thank you, Guy. So I guess there is a comment.
Hey, Guy.
Hi.
So yeah, it's great that you start to compare things with CW. I just want to point out this is not CW.
Yeah.
If you try the noise equivalent power of a frequency domain device, even if you just look at the intensity, is so much worse than a simple CW device. So I want that to be understood, that with a pure CW system, the SNR is way, way better.
Yeah.
And the other thing, will you try comparison with time domain next?
If someone has to spare money to give us the device, but no, yeah, I completely appreciate. I completely appreciate what you said about that, and yeah. So when I say CW, it's the amplitude from our frequency domain measurement. So it's not strictly, it's not true continuous wave. But I guess with the hardware we're using, yeah, those are just the things that we're measuring. And I guess ideally you could have like a separate true CW device and put the source and detectors like next to each other to truly compare. So yeah, so don't go away spouting that this, yeah.
So thanks. Regarding time domain, I guess that since you said that multi-modulation frequency is better than single, then time domain should be better because it's an infinite number of modulation, or not?
Yeah, well, I mean, I've never used a time domain device, so I can't say, but yeah, perhaps it is, but
maybe we can check if you come to Milan.
in a few years, we'll see.
Other questions? See, Idron? Quick question. We have 1 minute.
Just a comment to this effect. You can always use the Fourier transform. So we have often done it when we did time domain measurements and Fourier transform, then you can apply phase methods and vice versa. And there's one point, at least for homogeneous medium, you should be careful with the phase because it's just frequency multiplied by mean time of flight. So as long as you are homogeneous, you don't get more information when you use multiple frequencies, I guess. Experts are there. Thank you.
We should have a chat afterwards. Yeah, thank you.
Yeah, let's thank Guy one more time.
Thank you.
Okay, so for our next speaker, Rebecca Brad couldn't be with us today, so instead it's going to be Lorenzo Spinelli, and he'll be talking to us about a fast multi-channel time domain system for steady cerebral oscillations.
So, sorry. Okay, good afternoon, everybody. I'm Lorenzo Spinelli, and I give this talk on behalf of Rebecca that cannot be here today. This work was made with my collaborator, my colleagues of Polytechnic of Milan, Alessandro, Davide, and Rebecca. I will try to show you our time domain NIRS instrument that we devise for study the brain cerebral oscillations. So this is the outline of the talk. After a very brief introduction, I describe you our devices and also its validation. Then I tell you about the preliminary results of a numerical simulation we perform to understand where we can go with our instrument in the field of frequency of oscillation in the brain. Then I show you result on the very first in vivo pilot study and give you some conclusion and perspectives. So as the introduction, it is known that there is a periodic variation in the concentration of oxy and deoxyhemoglobin in the brain, and not only the brain. And these variations are due to some physiological phenomena and were detected, for example, with the MRI, but also with NIRS. These periodic variations are characterized by specific frequency or band of frequency. I reported here some example, and now we want to see if we are able to also measure the power, these power frequency spectra, also with the time domain instrumentation. Because we want to go to time domain, you know that I think all the basic principle of time domain, where we consider a light pulse injecting into the tissue, and then we recollect light emerging some centimeter far from the injections, and the distribution of time of of photons is affected in different way by the absorption and scattering phenomena. So we can decouple the two coefficients quite easily. And this is one of the advantages of use time domain. The other advantage is the depth selectivity of this technique, because time naturally encodes the depth information. The longer the photons travel into the tissue, the deeper they probe inside the tissue. So this is the schematic of our instrument. As a source, we had two high power pulsed diode lasers that are combined in order to get a time multiplexing approach. And then the light is shined on, in this way is shined, the two wavelengths are shined on the sample. On the detection side, we have a 4 hybrid called PMT, which are connected to 4 channels of time digital converters that give us the time resolution. All the instrument is controlled by a PC and is set into a rack with a modular arrangement. We validate this instrument with the now usual protocols of best, the BIP protocol and also the MEDFOT protocol. For example, we have in this case for the instrument 2.5 milliwatt on the sample for the red wavelength and almost 10 milliwatt for the infrared wavelength, and we have a width of the pulse of about 250 picosecond. Then we also performed the MEDFOT protocols on the MEDFOT kit phantoms, but I think most of you know these protocols. I want to puntualize and give you particularly the results of this characterization, because if you want to have a good power spectral density, we need a high sampling frequency. So we have to be able to perform measurement fast, and we can go fast, but we also need to have a good retrievement of the optical parameters mu A and mu S. So we consider this measurement on the phantom with the interfiber distance of 3 mm fixed and the constant count rate quite high of 7 millions of counts per second. We made a different acquisition with the acquisition time going from 1 second to 1 millisecond. And obviously, when you consider acquisition time short, you increase the noise of the retrieved parameter. But we found that at the end we can go at 10 milliseconds of acquisition time, that is a sampling rate of 100 Hertz, having a coefficient of variation on mu A of about or less around 2%. And this is quite good. Now I want to describe you a technical problem we observed, somehow unexpected, because from a measurement on a static and homogeneous phantom, we observed that in the power spectrum of the number of counts, we found these peaks, some peaks in frequency that are totally unjustified for this sample. We play around a little bit to understand which is the problem. And we also, quindi measure, we then measurement other, we test other frequency, acquisition frequency, and you can see that we have the frequency, the harmonics, and also the alias frequencies inside the spectrum. So a lot of peaks that we don't want. And at the end, the cause was the hardware reconstruction of the distribution of the time of flight histograms, which are constructed starting from a trigger signal. We try to change also this external, trigger signal, but the problem remains. Changing the external trigger signal changes the frequency, so the periodicity. And you see here that the photon counts have a clear, in this case, periodic behavior, and it repeats every about 6 seconds. And at the end, we solve this problem by exploiting the time tagger capability of our hardware. And then we record the stream of the photons arriving, and from the time tag of the photos, we reconstruct the distribution of time of flight in post-process. In this way, we don't have any more peaks in the power spectrum or modulation in the timelines. Then we also perform a numerical simulation in order to be to test if in time domain we could exploit the characteristic of be sensitive to the deep. So we consider a bilayer geometry. We use 2 interfiber distances. We also vary the number of counts per curve. And we simulate oscillation in oxy and desoxy background with an amplitude of 1% and a frequency of 1 Hertz. And simulate also acquisition of 50 minutes with an acquisition frequency of 20 Hertz. For the analysis, we use a mean partial pattern method from which we can get the variation of absorption in the up layer and the lower layer. And from the variation of absorption and also the absorption, we get, and the background absorption that we know, we get the oxy and desoxy concentration in the two regions. Just 2 results. We see that if the oscillation affect the upper part, we get these beautiful peaks around 1 Hertz, as we expect, only in the upper layer. In the down layer, we get nothing, no peaks. And also, if the oscillation affect the lower layer, we get the peak in that layer in the oxy and desoxy, okay, around obviously to 1 Hertz, that was the frequency we simulated. Then it seems now that we can go in depth, and we try to measure in vivo and in vivo subject. Sorry. And so we perform a 5 minutes resting state experiment with the subject lying on the bed tilted over 30 degrees. We consider an acquisition of 20 Hertz with a 4 centimeter interfiber distance, and we put, as you can see, the probes, the optodes on the forehead of the subject. We have also other physiological sensors such as ECG, breath sensor, breath strip, and other blood volume pulse, skin conductance, and temperature. And in the following, I will show you only the BVP and breath sensor results. This is just a very preliminary measurement, so just to see if we are able to recover the power spectrum. Here you can see the region of frequency of the power spectrum we measured around 1 1 Hertz, where you can see in green the peak due to the heartbeat. And we can see that also in the black line, both for the heart, we can see in the black line the same peak of both in the oxy and desoxy concentration in the upper layer. In the lower layer, there are no evidence of the presence of these peaks. Furthermore, we also inspect the very lower frequency band, and also we get that result that I underline that are very, very preliminary. Also, we can see the respiration peak around 0.15 Hertz in the oxyhemoglobin of the upper layer, of the down layer in this case, sorry. Okay, thank you. Okay. We also try to see if we are sensitive to changes. So we, after the measurement with the tilted bed, we put the bed horizontal, and some changes in the lower spectrum of the frequency are evidence. But these results are to be confirmed and are very preliminary. In conclusion, we realize a TD-NIRS systems or device for fast acquisition. We can go up to 100 Hertz with the suitable coefficient of variations. And from preliminary numerical simulation, we get promising results about the decoupling of oscillation in between extracellular and cellular tissues, that is upper layer and lower layer. And we get from simulation suggestion for measurement protocols and data analysis procedures. And I show you the first in vivo measurement on an healthy subject. As for the future, we want to increase the numerical simulation for the refinement of the data analysis procedure. And now is ongoing an experiment on 15 healthy subjects, consider resting state protocol and also forced brain acquisitions. Thank you for your attention.
Yeah, thank you for the very interesting talk. Maybe time for one question. One question I have, it's very impressive. Seems like you also have to store a lot of data if you're like time tagging the photons and going up to 100 Hertz. And like, how do you handle like the data storage, like for that kind of volume?
It was very huge files we had to manage because 15 minutes of acquisition is very, very long.
Yeah.
Okay. But we resolved. We was able to reconstruct the distribution time of flight. It's not difficult. We have also the plus that we can decide in post-processing at the acquisition time.
Okay. Well, thanks so much. Let's thank Lorenzo one more time.
Thanks.
Yeah, and then our next speaker will be Mitchell Robinson, and he'll be telling us about some exciting advances with DCS instrumentation.
Oh, they have to switch it. Okay. Awesome. Brilliant. I'm Mitchell Robinson. I'm working at the Martino Center at MGH. Today I'm going to talk about some of the advances we're making in long wavelength diffuse correlation spectroscopy for high SNR measurements of cerebral blood flow. So as the audience is aware, and as Ashwin gave a really, really good introduction to DCS before, I don't have to go too in depth, but just for a quick overview, DCS uses long coherence length light to measure the fluctuation of intensity of a single speckle. And so what I'm showing here is the vibrating red blood cells in your vasculature that creates a time varying speckle pattern on the surface of the tissue. And then we sample just that small square, and that traces the intensity over time. the rate that that intensity signal is changing is related to the speed of the red blood cells in the tissue, and we quantify that with the autocorrelation function G2. And then faster flow is a shorter decorrelation time, slower flow is a longer decorrelation time, and then with models, we're able to get what we really want out of the system, and that's the blood flow index, which is related to that, the blood flow in tissue. And so as a complement to NIRS, DCS has been shown to be helpful in distinguishing effects of traumatic brain injury. We can look at things like autoregulation and how blood flow instability can lead to interventricular hemorrhage in preterm infants. It's also been shown that it could be used as an early biomarker for Alzheimer's disease, and that the vasculature is dysfunctional. And then as it relates to the functional community, it's been shown that the hemoglobin-dependent signal is smaller than the CBF-dependent signal. And so if we can make DCS higher SNR so that we can get these signals out, it might provide a more sensitive measurement to cerebral activity. A problem with DCS is that extracerebral thickness generally limits its usability in adults. And so in newborns and adolescents, the extracerebral thickness is usually no more than 10 millimeters. And that's it allows for reasonable SNR with the standard DCS instrumentation. As we go into adulthood, that distance becomes much larger. And if we compare some results from a recent simulation study, for a typical 25 millimeter source detector separation, we can see that DCS doesn't provide sufficient cerebral sensitivity for many of the cases across the head. So we say 20% is a benchmark. It's just kind of a number here, but we can see that there's a lot of positions in the head that are not covered well with DCS. And in the same way with NIRS, you can go to longer source detector separations, but that is a trade-off in SNR. And so if we want to analyze how well DCS works relative to itself and other sorts of implementations, we can look at Monte Carlo simulation. And so the thing we did is we took a 3-layer model, set it up with optical properties, thicknesses, and blood flow index for that particular value. And then we ran 2 sets of simulations to look at sensitivity and then also SNR. So we have a baseline case where everything's flat, and then a second set of simulations where we increase the blood flow in the deep layer, which allows us to derive parameters like the relative BFI, which is our extracted change. Cerebral sensitivity is then defined as the ratio of how much of a change we extract versus how much we actually changed in the simulation. Because we have the flux of light from the Monte Carlo simulation, we can calculate the noise that we would expect to see on the correlation function, and then it allows us to calculate a coefficient of variation of our fit BFI. And then finally, we can define a contrast to noise ratio as the ratio of cerebral sensitivity and coefficient of variation. So it's more of an index of contrast to noise because we don't actually simulate a change, but we're just looking at how much of a change we can extract. And so if we look at how a typical DCS instrument works, 850 nanometers, 4 SPAD channels, we can see that for a lot of the separations that we have, we don't really have as good a performance as what we might want to have. And so our work is pushing toward finding ways to improve that performance and move up on this graph to have higher contrast to noise ratio. And so the first way that we're pushing is to use 1064 nanometers. And so it's not a very good wavelength for NIRS. You don't really have good contrast between oxy and deoxy at this wavelength, but because we're not looking at hemoglobin, we're looking at scattering contrast. It's definitely 100% fine just to move to the longer wavelength. And so when we do, we can take advantage of a lot of different factors that improve our signal to noise ratio. The first being just regulatory in nature. So based on the ANSI standard, we can put in 2.5 times more power. So that's 2.5 more times more photons back at our detector. Really, really great improvement to SNR, almost for free there. In terms of physiology, the attenuation at 1064 is lower than it is in the NIRS, typical NIRS wavelength range. And we also have an improvement on the shot noise characteristics. So because the 1064 photons carry less energy, per unit power, we see more photons for the same power, which allows us to have a better shot noise characteristics. We're having more observations of those counts. Combining all those factors, all other things being equal, same detectors, we would see about 11 times more photons. And in addition, the autocorrelation decay is slower at 1064, and so we get a better ability to fit that curve, and that gives us an extra benefit. Like I said, this is all detectors being equal. Unfortunately, we can't just switch from silicon SPADs over to InGaAs SPADs because the InGaAs SPADs don't have adequate performance, and so we have to think, okay, how do we still take advantage of the wavelength, but then move to detectors that aren't, that noisy. And so the approach we've decided on, and a lot of other groups have as well, is to use an interferometer. So we call this interferometric DCS. So I'm showing the basic DCS setup. And so our detected intensity is just dependent on what's coming from the tissue, that's E sub S, and then our autocorrelation function reflects that as well. The approach then is to add an interferometer setup. So you have a reference arm now that you're adding. Our math changes a little bit. And so we have this autocorrelation function now, which is a sum of a quadratic term for G1 and a linear term of G1. And if we compare what our signal to noise ratio of the curves looks like, what we find is that this is just in a SPAD, single SPAD, so we're not changing anything about the detector yet. We see an improvement in the signal to noise ratio of the curve. And if we look at the coefficient of variation with respect to how much reference arm we're using, we can see up to about 80% reduction in the coefficient of variation just by using this interferometric approach. And so that works irrespective of what wavelength you're using. And so we think this is a really nice approach just to take the typical DCS system and sort of elevate the performance to allow for longer source detector separations. And so what we're going to show here is when we combine these two approaches, we see a little bit better results. So they add well together. So if we go back to the graph, just using DCS with super expensive superconducting nanowire detectors, 95% of detection efficiency, really, really great. You get a nice improvement in signal noise ratio. And then if you take the interferometric approach as well, you improve your signal noise even further than that. And so in this study, we compared the performance of typical SNSPD DCS with our InGaAs lines camera IDCS. And our instrumentation will look something like this. So we start with our laser source. We split that between a reference arm and a sample arm. Here we take the 10% of the split and take that to a fiber amplifier and amplify to 200 milliwatts. And so that's one of the engineering benefits we have at 1064 is to take advantage of that communication sort of hardware that exists. So we can amplify to almost any arbitrary level, and we use that to allow for a dual source probe. So now instead of having a single source, we're able to use two sources, double the SNR. The footprint of our probe doesn't grow that much, and so it's a nice way to sort of scale that SNR again. In this probe, we also have a 5mm channel to look at short separation and scalp blood flow. 25mm channel, won't talk very much about that, and then a 35mm channel. And so in the 35mm, we have two sets of fibers. Oh, oops, jumped there. Okay, so we measured the 5mm with a silicon SPAD to get the scalp dynamics. It's like 3% efficient photon detection efficiency, so it's okay to see the short channel. At the long channel, we have the interferometer, so we have a multi-mode fiber bundle, and we project that onto the line scan camera. So we have 2048 pixels. We sample that at anywhere between 300 to 400 kilolines per second. And this allows us to sample approximately 500 independent speckled channels. At that long channel, we're also going to compare to our superconducting nanowire detectors. So we have 4 channels there. And because we have such a single probe, we're not really going to try to do as much functional experiment with this study, but we're going to look at more wider range physiological manipulations of blood flow. So we're looking at a hyperventilation task, which we expect to see a biphasic response where we first see the drop in blood flow and an increase. Pressure modulation, which is commonly used to look at the scalp sensitivity of a particular diffuse optical technique, and then breath holding, which we've seen to have a reliable increase in both blood pressure and blood flow. And so we'll see how that looks with both of our instruments. And so the first thing we wanted to do was to compare the performance. And so we wanted to look at how can we resolve the pulsatile signal. And so we sampled our signals at 100 hertz from both the long wavelength channels, and these are the results from 5 subjects. So in purple we have the SNSPD, and then in the green we have the InGaAs line scan results. And we can see by eye that the line scan camera is giving us a lower variability. And if we quantify that just by looking at the coefficient of variation for each of the beats, that gives us about a 1.7 times improvement in coefficient of variation just going from the SNSPD to this IDCS line scan system. And so you might ask, I mentioned earlier that you can get a 4 times improvement if you just switch from regular DCS to IDCS, but what this allows for is an approximately 7 times cost reduction. So these SNSPD detectors are quite expensive, and they're difficult to move into the clinic where we're trying to move these technologies. And so by allowing something that's about the same size as a typical DCS instrument, we can effectively measure as well as the bigger bulky system. And so we really like that. And then finally, we can look at how our subject average response and our physiological manipulations look. So in our hyperventilation task, we see the expected drop in blood flow and then the subsequent return to baseline. And we see that we have reasonable agreement between our long separation measurements. In the pressure modulation, we can see the benefit of going to 3.5 centimeters in that we have about 50% sensitivity to scalp, which is quite a nice improvement over shorter separation channels that are commonly used. We see a little bit of discrepancy between the two long separation channels, and so we're still kind of investigating what that might look like or why that might be. And then we had our breath holding task where we see that all of our channels are increasing in flow, but we do have that little bit of discrepancy between our green and our purple curves, and we're trying to figure out exactly why that's happening. But we can expect, we're excited that the SNR of the system is quite high. And so in conclusion, in this work, we compared two high SNR DCS methods and showed that the use of heterodyne detection can improve your signal noise ratio as well as your performance per dollar metric. We did see that discrepancy in the fit blood flow index, and so we're investigating why that might be. It might be the sampling rate is too low to really resolve the upper part of the curve, and so we just need to look more into what we're seeing there. And then future directions includes the use of multi-layer modeling to separate scalp and deep blood flow, and we have a pretty high confidence that because we're measuring at such a long separation, we'll have good performance there in the separability. And then we plan to adjust this design to allow for multi-channel operation. And with that, I will plug all of the awesome posters from the group at Martinos, which are scattered across yesterday, today, and tomorrow. And then thank you so much for your attention.
Yeah, thank you, Mitchell. It's a very exciting talk. Yeah, any questions?
Hi, Mitchell. Do you think that there'll be sensitivity to the motion, given it's an interferometric system? Will it be sensitive to fiber motion itself? Have you tried wiggling the fibers? Does that cause anything to happen?
It seems reasonably tolerant. I didn't show results here. We had a few subjects do sort of a head shaking task to see how much motion you might expect, and within reason, it seems to be motion tolerant. I think you had to get like very big shakes to get it to start to have issues.
Yeah, yeah, any other questions?
Thanks for the nice talk. I was wondering if you could apply this approach also to shorter wavelengths. I mean, maybe you gain something also without going to longer wavelengths.
Yeah, exactly. So the Srinivasan group, I guess now at NYU, has a line scan camera system at 850 nanometers and sees the similar improvement to DCS signals. And I think they showed autocorrelation curves at like 5 centimeters or something like that when you average long enough. And so it's pretty exciting. This approach of multi-speckle plus interferometric detection.
Another question. Is there any difference in terms of depth sensitivity with the interferometric approach as compared to the classical DCS approach?
We don't think so. It's the same sample light, and it's just shifting the curve. There might be a numerical difference in the fitting just by some funny metric, but the light that you're measuring is exactly the same. You're just measuring a G1 representation instead of a G2 representation.
Thank you. Thank the speaker again. Okay, we are arrived at the last talk of our session, given by Xiaojun Chen. I hope I pronounced correctly. Development of a fiber-based spectral contrast optical spectroscopy system to measure human cerebral blood flow.
Okay, hello everyone. It's my pleasure to be here to talk about our development of a fiber-based gas system to measure human cerebral blood flow. I'm so happy you've already heard about speckle patterns like this in the first talk. So you already have some idea of what speckle contrast is, but let's still have a brief overview of the speckle contrast stuff. Like for optical methods for human brain function measurements, if we distinguish the technologies from their light source that they're using, we can group them as two groups. One is those that use incoherent light source, which is fNIRS that we have been hearing about all day. And the second group of technology that use coherent light source are the speckle dynamic-based technologies that can be used to measure blood flow. And within this group, we also have two different kinds of technologies. One is that use the temporal statistics of the speckle patterns, and another use spatial statistics. For temporal statistics, which is DCS, you've also heard about it. What we do is we look at the speckle pattern evolution over time, and you pick a point or you pick one speckle, and you look at how the speckle intensity fluctuates over time. And what we measure is the intensity autocorrelation function of that speckle pattern. For the spatial statistics, instead of looking at a series of speckle patterns, we look at like one speckle pattern at a particular camera frame for a particular camera exposure time, and we calculate the contrast, which is defined as the standard deviation of the intensity values here divided by the average of the intensity values. And as you know, these two technologies are related, and the contrast that we calculated here is directly related to the G2 that we calculated in DCS. Okay, so why SCAS? I mentioned several technologies, but not SCAS yet. For DCS, we know it measures human CBF is great, but we also know it has relatively low SNR and also use like relatively more expensive detectors, single photon counting device. Laser speckle contrast imaging, which is great, but the traditional laser speckle contrast imaging is used to provide 2D maps of CBF in small animals. Like this, here are examples of the wide field imaging of mouse brain from the raw speckle pattern to the speckle contrast imaging of blood flow maps. This has high resolution, high speed, inexpensive, but it requires thin skull or craniotomy preparations, which apparently we don't want to do that for human beings. So that's where fiber-based SCAS that comes into play. So it has all these advantages, has high temporal resolution, inexpensive, a high SNR, but because its technology is still at its very initial stage of the development, so there are many things we still need to resolve. For example, we need to construct a system. We need to develop a noise model to predict its performance. We also need to develop the data analysis pipeline to obtain a quantitative blood flow value. Okay, so here is like the schematic of our fiber-based SCAS. We have a laser come in at 850 nanometer, goes through a fiber into a scattering sample, and use another fiber to collect the light and image onto a CMOS camera. On the source side, we have utilized the pulsing strategy to improve the photon flux within the camera exposure time. What we do here is that you see we use a chopper wheel so that we reduce the duty cycle of the laser to 10%, such that we can increase the photon flux by to 10 times within the camera exposure time, while keep the average incident power to be still within the safety limit. On the detection side, right now what we have designed to image two custom-made rectangular shaped fiber bundles onto the same camera. And here are the examples of the images of the fiber bundles when they are focused and when they are slightly defocused. On the human subject side, like we have engineered the source and detector optodes, and they can be, they fit our 3D printed head cap by a spring top over here. And this is how it looks on a human subject head. And this is one example of the raw speckle image from human head measurement. I mentioned that we need like modeling to determine the performance of SCAS. We have developed a model for a noise model for SCAS. I'm not going to talk in details here. Shari had a poster today. I hope you had a chance to talk with her describing our noise model. But if you missed it, you can still find her or talk with me about the details. But the take-home message is, using the noise model, we have compared the performance of SCAS and DCS. And here the result is we can, for the 2 cameras of SCAS, as compared to a 1064 channel SPAD camera using DCS, we can achieve about 28 and 19 fold of improvement in terms of contrast noise ratio compared to DCS. Okay. And tomorrow, Stefan is going to talk about like their work of using this noise model, the prediction of the contrast noise ratio. Here, they compare the SCAS, use the Hamamatsu camera to the single channel DCS. I mean, this is traditional single channel DCS, not the fancy DCS system that Mitchell just talked about. And here, from here, we see 2 orders of magnitude improvement of the pulse DCS, pulse SCAS that I've talked about the pulsing strategy before, as compared to the traditional single channel DCS. Okay, so that's great, because our numerical results show that SCAS can outperform DCS by at least 10 times. For here, we put a criteria here for an equal or lower total price of the system, because we want to make a fair comparison. For example, here I have a single channel DCS, but I mean, if you're rich enough, you can also make a million channel DCS to average over, to improve your contrast noise ratio, but that is not feasible, okay? So the 10 times improvement, it sounds a little hard to believe, but this is mainly because in SCAS, we use a CMOS camera that has millions of pixels. So you have millions of pixels or millions of speckles that you can use to average to improve the CNR, okay? But there's no free lunch. There are challenges to SCAS. What we calculate the contrast, there will be noise that's contributing to the standard deviation of the intensity sets. It will add bias to the contrast that we measured. So we have constructed this data analysis pipeline to reduce the bias that's being added to the contrast due to noise. Mainly coming from shot noise here and ring noise. I will give you some idea like how much bias it adds to. Here's the simulation results we have for like a Hamamatsu camera with the properties here. You see the difference between the magenta, which is the we call the fundamental contrast that's related to the blood flow, as compared to the red curve, which is the total raw contrast you measured from the speckle images. And here, another interesting thing is you can see we can reach a shot noise limit. You see, like when you compare the black line and the blue line, which is shot and ring noise, we see like if we integrate longer, we can reach the shot noise limit without the, I would say, the complicated like implementation of interferometry and such. And at a higher photon flux, we see it gets into the shorter shot noise limit easier, and also the difference is smaller. But for like a camera that has a larger ring noise, it's harder to get shot noise limit. And another example. Okay, so now I have already described the data analysis of noise removal. I'm gonna summarize our data analysis pipeline. What we do is first obtain the raw image from the camera. We subtract the dark image when we turn off the laser. And then we select the ROI, because I told you like now we image 2 fiber bundles onto the same camera. Let's first select one of the fiber bundle. And then we calculate the contrast, raw contrast within every 7x7 window. So we get an image like this. And then for every 7x7, for every like pixel here in this image, for every 7x7 window, we subtract the contribution of noise, the bias that's being added by noise, shot and ring noise removal. And then we average over all of these pixels, we get a single number of fundamental contrast for every camera frame, okay? So I talked about this noise correction stuff, but we have to, this requires accurate measurement of the shot noise or camera gain or and camera read noise. We need to figure out a way to make sure that what we're doing is correct. So what we do is like we validate this noise correction scheme using LED light or incoherent light. I told you like in the very beginning, those speckle dynamic space techniques, they need coherent lights. So if we have LED that's coming to the sample, we're not going to measure any speckle pattern. So our expected K-squared, the contrast square, should be 0 after we do this noise correction. Okay, so here's one example of the correction. You see here, even when we use the LED light, we still see the blue curve is before the noise correction. You still see like a little bit cardiac when we measure on the human forehead. This is because due to the shot noise contribution. But after correction, we can bring it down, the level, bring it down to like below 10 to the minus 4. And our talented student, Kenny, he had a poster today. He talked more details about all the correction scheme, and we are able to bring down the remaining K-square using LED light to be more than 1 order of magnitude smaller than the contrast that's induced by blood flow when we are using laser light. So we are happy about this, but of course we can further refine this noise correction scheme by more careful measurements of the camera gain, ring noise, and other camera properties. Okay, so now it's the fun part, our preliminary human brain measurements. First, I'm going to show the cardiac pulse waveforms that we measure on the human forehead at a source detector separation of 30 millimeter with a resolution of 46 hertz. Here's the camera counts. I show here both the results for CW, the continuous wave laser light source, or and that we have for that we have included that pulsing strategy to improve the photon flux. And here's the result for blood flow or 1/over blood flow because we measure contrast. We are very happy to see this very clear. You see cardiac pulse shape at this large source detector separation of 30 millimeter and at a very high frequency. Because previously we have not seen this high quality measurements at this large source detector separation in our traditional DCS at a 50 nanometers before. Okay. We have also conducted preliminary human brain function measurements. What we do is like a simple mental subtraction task. The subject is asked to subtract a small number from a large number. You just keep subtracting until we tell you to stop. And here is the evoked response for the subject. We see that K-square decreases or blood flow, because blood flow is the inverse of the K-square, increases by 10% during a stimulus, and then it comes back to baseline as expected. What is nice is here, we also see the camera count, or you just directly measure intensity. It also drops by about 2%. Because this is basically the single wavelength fNIRS measurement that we can do. Okay, so here, summary, we have developed a fiber-based gas system to measure human cerebral blood flow. And in the future, we will recruit more subjects and quantify the performance of SCAS experimentally. Hope to show the more than 1 order of magnitude improvement as compared to DCS. Okay. So my last slide, I want to thank everybody in the BOAS group and all our collaborators. I want to thank our funding support from Meta Platforms. As the last talk of today, I also want to thank the organizers of this amazing conference. I'm so happy to join this lovely community. And also thank you for your attention.
Thank you. Now the talk is open to comments and questions. This is the last talk, so if we do comments, then we go to the dinner.
This might be a boring question, but what was the frame rate that you were using for those in vivo measurements?
For this, now it's 46 hertz. Actually, we can go higher. It's not like we can go maybe, oh, the slide is no longer here, but yeah, it's 46 hertz for now. It's because I can tell you, like we had a funny way to slave our camera to some synchronization signal that actually reduce our camera frame rate. But the camera actually can go up to like 89 hertz. So that's not the limitation of the technology. It's like our, way of when we start this.
And do you leave the laser source on continuously, or do you like pulse it?
We pulse it.
So you like clean the camera frame, then turn the laser on? then turn the
Right.
laser off, and then
You-
do the
Right, not on
readout?
and off, but we use like a chopper wheel blade. So the laser on is all is kind of, you know, on the laser side, it's always on, but the chopper wheel, it like cuts off the laser like 90% of the time. So we use that to pulse the laser, because actually we also try to pulse the laser directly using the control of the laser, the control box of the laser. But then we see there's some actually reduced like coherence due to that. So we don't want to do that.
So is that chopper wheels are inconvenient? Like, I don't know. Maybe chopper wheels are fine. I'm thinking if you want to multi-channel this so that you could do like a tomography system, but
yeah.
And so that was 2 detectors on 1 camera.
Yeah.
So that's still $5,000 a detector.
You mean the detector?
Or I don't know, how much does a camera cost? 10,000?
It's around that. Like,
Yeah.
but we can also use like lower cost cameras. That's why because like there's, of course, in the beginning we want to use better cameras because we want to make sure all our noise correct and things like to work. But when we use like lower cost cameras, like $500, but you know, we still need to do
So
more
you
things.
don't think you need really low readout noise?
If we can do all our, you know, noise correction correctly, of course, if we have low read noise, that's good. But like a few things we can deal with that. For example, we can increase right now. Like the simplest thing we do is like we can increase our exposure time. The previous slides we showed, if we keep increase our exposure time, we will reach the Shaw noise regime. So we no longer like be limited by read noise. But of course, we cannot integrate it too long because the contrast itself also decrease. Then we'll have some digitization errors like this. Yeah, it's a tricky thing. Like we're investigating. Yeah, thanks for the.
Yeah, thanks.
Yeah.
Thanks to speaker again. Do not leave the room because we have to make an announcement. Okay. It's open. Okay, so this is the map to reach the Royale. It's about 45 minutes. It says 53, but if you just speed up, I think you are hungry. And it is at 7:30, if I remember well. Okay. So you can also take the tube or the Uber, whatever you want. So see you there. Thanks all the speaker and all the participants. Yeah, do not forget the ticket for the entrance and the ticket for the drinks.